Smart University. Sorry. Continues to grow. As an international university, possessing the values of academic freedom, dynamism, transparency, flexibility, and innovation, the main objective of it is to expand its influence from local to global. To achieve this objective, the university is increasing its academic and social activities by organizing national and international congresses, striving to meeting international academic standards and developing high-quality international strategic partnership. Firstly, I would like to sincerely thank United States Department of Agriculture, Foreign Agriculture Service, and our Department of Molecular Biology and Genetics for their efforts and contributions in organizing this meeting. Their efforts in the organization and execution of this significant scientific platform are valuable for the advancement of science and enlightenment of society. As we know, Turkey has significant potential for agriculture due to its geographic location, climate diversity, and rich biodiversity. Agriculture is not only a vital sector for Turkey's economy, but also an integral part of the social and cultural structure of our society. Therefore, closely following all new technological advancements in agriculture and plant breeding and integrating these advancements into our practices are of great importance to us. The development and expansion of international collaborations play a major role in the application of agricultural and plant breeding technologies. Developing these collaborations encourages knowledge and experience sharing and is necessary for us to develop global solutions. Especially, new technologies developed in plant breeding will play a significant role in vital areas such as food supply and safety, human health, and energy resource. These developed technologies will provide solutions to environmental and social challenges we will face in the future and will aid in creating sustainable future. Çanakkale 18 Mart University. We take great pride in taking a leading role in such scientific meetings and seminars. Our active participation in the introduction and dissemination of innovative technologies is a crucial factor in our society's understanding and its acceptance of these innovations. These platforms, where information and experience are shared, will facilitate the better communication of technological advancement to the whole society. I sincerely thank everyone who contribute to the realization of this significant meeting. I hope this meeting will provide valuable international to its participants and will be a productive experience for everyone. I wish that we all depart from this event with new perspective, ideas, and solutions. On this occasion, I wish everyone a good meeting and a good day.
I extend my appreciation to our Vice Rector for her warm welcome and insightful remarks. Thank you. Now, I invite Michael Frankum, Agricultural Counselor, coming from Ankara.
Yes, thank you for that introduction, and thank you, Professor Evren, for those opening remarks. I think they were very fitting. On behalf of the U.S. Department of Agriculture and the U.S. Embassy, I'd like to welcome each of you to today's special event. I'm closely watching the number of participants tick upward and I'm getting excited as it approaches 100. But today we have government officials, we have academics, scholars, private sector, we have colleagues from my agency back in Washington and perhaps a few others joining us. We also are joined by our invitational speakers, Dr. Catavelli and Dr. Thompson, Dr. Tuna as well from TÜBİTAK, and we're really excited to have the three of you speak to us. And of course, I would be remiss not to mention Dr. Tashkin. He's been a very close partner of ours over the last number of years. We've appreciated working with him and his colleagues and his university, Çanakkale University, in helping build connections and linkages with US institutions and helping in this exchange of information. I'd also just want to say thank you to the other people on the call today, officials from the Ministry of Agriculture and other institutions such as TÜBİTAK and the close relationship that we have with them. We really value that and look forward to seeing that flourish in the future. This is the second time we're holding an event on this topic, which I believe helps really underscore the importance of this technology and making it accessible to not just researchers in the laboratory, but also to the farmers in the field, and making sure it has relevant applications to improve food security, not only here in Turkey, but elsewhere around the world. Today, we're going to hear perspectives from not just Turkey. We're going to hear from Dr. Thompson, who will talk to us about how this new plant breeding technology is being used in the United States. Dr. Catavelli will talk to us about his experience in Italy and the EU. And I hope these perspectives will be informative for you as we look to the future here in Turkey and orienting towards the adoption and full utilization of the diverse array of new plant breeding technologies that are out there. I think one thing that we'll hear today, a common thread that we'll find throughout all of the presentations, is these technologies, for example, gene editing, I think have real power to be an important game changer in agriculture throughout the world. It's not just this technology. Of course, there are other elements that need to be incorporated in the climate smart agricultural package to achieve the gains that we need, a second green revolution, if you will. But this is an important element of that overall package. What we need to see here in Turkey, I know for the last number of years, there's been discussions and conversations about the future of plant breeding in Turkey. And I think it's important that the regulatory door, if you will, open more widely to allow for the technology to be used. We need to get beyond just the theoretical and abstract application of some of these technologies, and we really need to see them in the field. And in this way, Turkey will be able to improve its capacity to produce agricultural products, not only to feed its own population, but to be an important supplier of food throughout the world and helping improve global food security. So I'm excited again to welcome everyone to today's conference. And in the short span of my remarks, I now see that we have 103 participants joining. That's great. I'm really excited. And as was mentioned by our opening speaker, I look forward to a productive conversation, one that will resonate and drive change. Thank you very much.
I appreciate the remarks that Michael Franken pointed out in his speech. Thank you so much. Next, we have Professor Dr. Kemal Tashkin from Çanakkale 18 Mart University, who will provide us with the scientific introduction and extend a warm welcome to all participants.
Hello, everyone. I just would like to thank everyone who involves in this project, especially thank you to USDA-FAS in Ankara office, Michael and Nergis. Your support is very important for us, and also would like to thank to our vice Chancellor, Professor Dr. Evren Karayel Gökkaya, for her welcoming and opening speech. And also, I would like to thank each speaker for sharing their valuable and important information with us today. Serkan Tuna, Luigi Cattivelli, and Michael J. Thompson. I don't want to lose much time because I have a presentation. I will directly proceed to presentation. I hope you can see my presentation now. And today, it might be better if I just, yes, sorry, just a minute. Stop sharing. Yeah, for the quality of this broadcast, I need to stop my video and share my screen instead. Sorry about that. Okay. Okay, now this one. I will be quick. Okay, I hope you can see my presentation now. Wonderful. So this is the outline for my presentation. As I told you before, I will be quick because I know everybody involved in this, participate in this event right now, already know this information, and just give you some brief information about plant breeding and new breeding technologies for introduction, of course. So plant breeding has been in practice for centuries. We already know, evolving from simple selection process to advanced genetic manipulation, what we employ today. But its main objective is the same: to alter traits to plants to produce desired characteristics. And one of the main primary goals of the plant breeding is to ensure food security by just enhancing crop yields, improving disease resistance, and enhancing quality. Plant breeders actually play a crucial role in meeting nutritional needs of growing global population. Additionally, plant breeding has implication for human health by enriching nutritional content of our food crops. We have the potential to increase right now levels of vitamins and other beneficial compounds that are very important for human. And also, I need to mention about climate change, since we already face this consequence of the climate change. Plant breeding becomes even more essential by just developing plant varieties that are resilient to changing climates, like changing in temperature, precipitations, salinity. We need to ensure that sustainability of our agriculture system, actually. Okay, next slide. Wonderful. And traditional plant breeding is a very old technique, involves careful selection and propagation process, carry out over successive generations. One of the key approach in traditional breeding is the crossing, or just hybridizing of different strains of variety of plants. With this technique, you can actually combine the genetic material of parent plants with desirable characteristics. So in this case, breeders aim to create offspring that inherit best traits from both parents, actually. And this traditional plant breeding primarily relies on phenotypic selection, involves in observing and measuring observable traits of plants. And these traits mostly, as you may already guess, are height and fruit size of the disease, fruit size or the disease resistance. We can give some examples. And in this slide, I just would like to, of course, without this slide, I cannot tell you about the plant breeding history, or and it's a very important slide that shows significant achievement in plant breeding, actually. We have archaeological evidence shows that domesticated seeds date back over 10,000 years. So you can imagine that as early humans just transitioned from hunting and gathering to farming, the initial step involves just recognizing useful plants and facilitating their growth while just discarding or avoiding non-useful plants actually. So these steps was very simple. They just collect the seeds from valuable plants, clear space and planting them, and go on. And of course, maybe it's better to talk about a little bit Mendel's law and discoveries and formulation of laws of heredity. That is a very significant major milestones in plant breeding. So this with the Mendel, actually we understood finally physical traits, connection between physical traits and genes. And then, of course, in the molecular age, particularly the cloning has enabled us to obtain transgenic plants, modification and gene editing in plants actually. And as you may already guess, before the molecular age, plant breeders manipulated genes and selected for specific traits without a clear understanding of the underlying genetic mechanisms. Plant breeding, of course, involves diverse array of technique, including selection of plants with desirable traits and propagation, and the utilization of advanced molecular methods right now. So another method employed in plant breeding is crossing. In this slide, I just would like to mention this one as well. And the crossing occurs when a breeder transfers pollen from one plant to the pistil to the sexually compatible plant. Eventually, they obtain a hybrid that carry genes from both parents' plants, actually. And of course, despite these natural breeding methods, rate of the mutation occurring naturally is often too slow. This will actually slow down plant breeding as well. So in this slide, I just would like to show you independent centers of domestication refer to specific region, and especially where the early humans cultivated and domesticated various plant species. These regions serve as epicenters for agricultural innovation in human civilization. So domestication is the process of selectively breeding plants to exhibit desirable traits. And these traits, as I told you before, include some characteristics like stronger central stem, reduced side branching, seeds that remain attached to the plant, synchronized flowering, fewer but larger fruits or seeds, and of course, we shouldn't forget the reduction in bitterness, actually. And we have archaeological evidence that shows us domestication of important crops like rice, maize, wheat, for example, took place over 6,000 years ago. And traditional plant breeding, as I told you before, in traditional plant breeding, natural genetic variation plays a crucial role to create new plant varieties. But this variation can only arise in a process what we already know of random mutations, actually. However, to expand genetic diversity further, breeders require inducible mutation. So intentionally induce variation using mutagenic agents. These agents can be like physical mutagens or chemicals in nature, EMS, for example, or X-ray irradiation. These are the very powerful techniques we use to obtain rice cultivars, wheat, oat, grapefruit, some other crop species. Examples are just given in here. And if you want to know more about them, you can actually visit this website, Mutant Variety Databases. You can learn more about them, actually. And another method I need to talk about, the hybridization, of course. the primary methods the breeders employ is just cross-pollination. They just aim to combine characteristics of two different plant varieties, of course. And again, they try to achieve desired traits like larger size or improve larger size fruits or improve disease resistance, actually. But as you may already know, these techniques require years of dedicated efforts, of course. And the hybrids are very important right now for our sustainability of our agriculture production. But it's also important to recognize the challenges associated with hybrid seed production, actually. So the subsequent generation, as you already know, may not be exhibit same vigor. This is the most important issue, maybe. And of course, I just would like to talk about Norman Borlaug and share his remarkable story with you. He's the extraordinary agronomist and humanitarian who actually forever changed the landscape of global food production. He also known as father of the Green Revolution. His work not only increased food production worldwide, but also play very important role to alleviate hunger and saving countless lives, actually. So he just developed high-yielding wheat varieties that changed farming practice forever. One of his key breakthrough was the creation of the dwarf strain of wheat with shorter stems. Unlike taller wheat varieties that would bend or break under the weight of fertilized heads, Borlaug just developed shorter stem wheat, remained sturdy. And this also increased the production level, yield, I should say. And I feel very lucky since I had a chance to meet him in a conference during my PhD studies in Italy, actually. And I try to make it simple and fast. Of course, I also need to talk about genetic engineering, also known as GA. It's a powerful technique that involves intentionally modifying organism genetic material. So, and I also need to talk about a little bit methods we employ in genetic engineering, like recombinant DNA techniques and transformation techniques, actually. But briefly, I should say that these are the terms: GA, GMO, transgenic plants, organism, biotech plants, organism, all LMO. These are the terms that we use or hear very frequently, actually. And they almost have the same meaning, but still there are some differences between, depends on who uses these terms, actually. The term living modified organism, for example, employed within the context of Cartagena Protocol on biosafety. So I believe you already hear about these terms very often. And what is GMO? This is also another term we frequently hear about it. And this term was defined in the EU directives. Like GMOs are defined as the organism apart from human beings whose genetic material has been altered in a way that does not occur naturally by mating and or natural recombination. We do also have almost same definition for the GM, and the genetic engineering, I believe you already know, that involves modification of plants by just adding specific genes or suppressing genes, for example, using RNA interference technology. What makes very interesting and very fascinating is this technology is the precision. So with genetic engineering techniques, we can actually introduce single segment of DNA with targeted precision. So let me continue why this technology is very important. This technology is very important, of course, but the reason behind this technology are quite similar those behind selective breeding we just mentioned in a previous slides, actually. We still wanted to enhance the nutritional value of the crops, increase their yield, make them more resistant to insects, for example, and we try to protect them against diseases. But using selective breeding that involves crossing plants repeatedly to identify desirable traits, genetic engineering allows us to introduce specific traits directly into the plant's DNA. You just bypass the limitations that observed in traditional breeding methods, actually. And yes, okay. And one common application of the genetic modification is enhancing resistance to pests. So what kind of plants, GM plants, they produced in worldwide? In this slide, I just would like to mention about the GM plants. So the genetic modification is enhancing resistance to pests. It's very important. Scientists actually introduce genes to obtain resistant plants to insects, actually. And with these techniques, you can also reduce the need of pesticides and minimize damage to the plants, actually. So you can also protect environment as well. Another important aspect, developing tolerance to the herbicides. And with the genetic modification, crops can be engineered to withstand with the effects of herbicides. And also another important issues, they employ enhanced crop resilience to the environmental stressors. So the scientists right now, since we are already faced with the climate change, they are developing crops that can withstand drought conditions. And in addition to stress tolerance, genetic modification also offers the opportunity to enhance the nutritional content of the crops, of course. One of the most and very well example in this case is the golden rice. Okay. Now I can talk about new breeding technologies. This is the exciting advancement in the field of plant breeding, of course, and the cutting edge technologies that change the way of approach we approach the crop improvement, of course. So with the new breeding technologies, we can now target specific genes or regions of the plant genome that will allow us to develop some plant varieties that you cannot imagine before, actually. One such technique is there are some examples in here I just would like to mention. And one example is the MES, marker assistant selection. And in this technique, we use genetic markers to identify desirable traits in plants. And with this technique, actually, we just speed up the breeding process by just helping breeders select plants with the desirable genetic makeup more efficiently. Another remarkable new breeding technologies we just mentioned actually in the previous slides is introducing genes from one organism into another to confer desired traits. And of course, most groundbreaking development in new breeding technologies is the genome editing technologies that we will hear more about these technologies in this event, actually. So molecular assistant selection MES depends on molecular markers, which are DNA sequences with specific characteristics, actually. So unlike environmental factors, these molecular markers, such as DNA markers, of course, are not influenced by growing condition or other external factors. And use of these molecular markers allow us to continuous observation of traits throughout different stage of plant growth. That's very good for us, actually. And different molecular techniques like RFLP, AFLP, SSR, rapid, and SMPs actually used to identify these genetic variations. And using these techniques, molecular techniques I just mentioned to you, we just reveal polymorphism. It means genetic differences in the marker alleles, of course. And let me go to the next slide. Of course, I just would like to talk about a little bit plant genome editing technology using site-directed nucleases or SDNs. So site-directed nucleases, we have few examples in here, like zinc finger nucleases, transcription activator like effector nucleases, and widely popular cluster regularly interspace short palindromic repeats associated nucleases. CRISPR-Cas9, actually. And this is the most well-known or widely used method for plant genetic engineering right now. I believe that our speakers will talk about them more details, will give you more details about these techniques for you. So I just don't want to spoil their presentation, of course, but just give you brief information what we can do with these diverse DNA modification technologies. We can just delete deletion of one or few DNA letters or even larger stretch of DNA. It's possible right now with these techniques. And this also allows us to selectively remove specific genetic sequences, and you can actually study function of the particular genes, identify their correct functions. Very important for us to know, actually, as a scientist, I mean, or just eliminate undesirable traits. And secondly, point mutation is also possible. It means that a single DNA letter is altered into another DNA letter. This is a precise alteration and have very important implications, such as it enables us to correct genetic defects or just introduce specific variation to increase enzyme activity, for example. And furthermore, addition of one or few DNA letters can also be achievable with these techniques. So you can actually introduce novel genetic sequences, including specific genes or regulatory elements, which are very important effect on the phenotype of the organism, actually. And also with the CRISPR-Cas technology, we can rewrite a stretch of DNA and offer many variations in the genome, actually. So synthetic biology, I just would like to mention synthetic biology a little bit because it's very important. It's coming, actually, it's very close. This technology is very new and very close. It's been around for a longer time, maybe, but still this technology offers us more interesting traits or developments. So synthetic biology definition can be can be found in different sources. In one definition, as it's written in here, it's a further development and new dimension of the modern biotechnology that combines science, technology, and engineering to facilitate and accelerate the understanding, design, redesign, manufacture, or modification of genetic material, living organism, and biological system. So in plant breeding, it means that you can actually engineer plants to produce valuable substances like biofuels or medicine. Or just you can, with these techniques, you can create crops with novel traits that are not possible through traditional breeding or genetic modification. And of course, the tools used in synthetic biology is very diverse, and some examples are given in here, like DNA synthesis. We can now synthesize DNA in test tubes. Of course, it's quite possible right now. This is one of the fundamental tools in synthetic biology. So you can actually customize DNA sequences and construct new functions, actually. And of course, next generation sequencing is also very important, and bioinformatics needs to analyze these data, and directed evolution, genome editing, engineered gene drives. Another important issue, we haven't discussed this one yet, but we need to discuss, I mean, in Turkey, we need to discuss this one, gene drives, very soon, I believe, and RNA-based tools and machine learning and other important tools that has been used in synthetic biology as well. Okay, I just skipped one slide, sorry. And of course, regulations, one of the most important and significant topics we discussed in the field of biotechnology. The global regulatory landscape for new breeding technologies varies widely. This also reflects different perspectives on genetically modified organisms, actually. So in contrast to uniform regulatory approach, certain regions have adopted a different approach by just distinguishing between different new breeding technologies based on process involved, actually. And the nature of the final product is also very important for decision. For example, they differentiate between techniques that introduce foreign DNA into plant is very important, and compared with the simply edit in the existing genetic material is different, completely different. So these are the different issues that global regulation address and answer, actually. And varying regulatory approach, the NBTs or new breeding technologies, have significant implication for the development of these techniques, deployment and accessibility as well. So these regulatory approaches is very important that we can take very seriously because it can shape the direction of research and development and eventually affect the availability of NBT-derived products and impact international trade as well. So as you can see, these regulations play a crucial role in determining how new breeding techniques are evaluated for safety, environmental impact, and commercial approval as well, actually, and public acceptance and other issues. We need to we need to reach public more efficiently, actually. Okay, my last slide actually is this is my last slide. I just would like to express my heartfelt gratitude to the United States Department of Agriculture, FAS, especially in Ankara office, and also Çanakkale 18 Mart University for organizing this virtual seminar. And also would like to thank to our esteemed speakers, Professor Luigi Catavelli and Dr. Serkan Tuna and Michael J. Thompson. I know they were very busy when we first contacted them, and they are now sharing their research and valuable insights and expertise with us. Thank you very much. And also would like to thank all the attendees, okay, in this seminar, and all the organizing committee as well, actually, and technical support as well. Thank you very much for joining us. I hope this event will be very useful for everyone, actually.
Thank you, Professor Dr. Kamil Taşkın, for your kind words and introduction. Before calling our first speaker, I would like to recall that there is a simultaneous translation in this seminar. From the menu below in the Zoom application, you can choose the Turkish translation and watch this seminar in Turkish. Yeni gelen arkadaşlarımıza hatırlatma yapmak istiyorum. Aşağıdaki menü seçeneğinden şu an anlık bir çeviri olmaktadır. Yayını aynı zamanda Türkçe'de izleyebilirsiniz. Tekrardan iyi seyirler diliyorum. Now, I'd like to call our first speaker. Our first speaker is Serkan Tuna, an expert of scientific programs from the Technological Research Council of Turkey. He will be speaking to TÜBİTAK's Research Support Program for Academia. Please feel free to ask questions in the chat section during the presentation, and the selected questions will be addressed during the Q&A section. Welcome.
Merhabalar, herkese teşekkür ederim. Öncelikle bu organizasyonu gerçekleştiren Çanakkale 18 Mart Üniversitesi'ne, USDA'ye, özellikle Nergiz Hanım'a ve Kemal Melih Hocam'a çok teşekkür ederim. Ben sunumumu Türkçe gerçekleştireceğim. Belki tercümanımızla bunu daha önceden konuşmadık ama mümkün olduğu kadar da yavaş anlatmaya çalışacağım. Tabii süremizde biraz sıkıntılar olabilir. Ben hızlı anlatmaya çalışacağım. Hedef kitlemiz biraz da Türkiye'deki akademisyenler olduğu için ve Ardev'in desteklerini belirteceğim için Türkçe bir sunum gerçekleştireceğim. Sunumun başında da size e-posta adresimi paylaşıyorum şu anda. Ekranı paylaştığım zaman göreceksiniz. Bir saniye rica ediyorum. Şu anda sanırım sunumum geldi. Burada da soru cevap kısmı yapabiliriz. Belki süreyi efektif kullanmak adına daha sonrasında bana e-posta da gönderebilirsiniz. Ardev'e dair, TÜBİTAK, TOVA'ya dair her türlü sorunuzu, destek programları ile ilgili seve seve cevaplarım. Teşekkürümü etmiştim. Bu arada küçük de bir düzeltme yapayım. Evet, az kaldı. 2 sene sonra doktor unvanını alacağım. Şu anda henüz o doktor unvanı yok ama en kısa zamanda almayı da istiyorum. Çalışmalarım devam ediyor. TÜBİTAK olarak bizim temel stratejimiz nitelikli bilgi ve nitelikli insan yetiştirmek. Bu nitelikli bilgiyi tabii ki destek programlarındaki projeler sayesinde nitelikli insanı da verdiğimiz burslarla sağlamaya çalışıyoruz. Bugün özellikle tarımsal biyoteknoloji alanındaki destek programlarından bahsedeceğim. Bol bol size istatistik vermeye çalışacağım. Sonrasında yani sadece proje destekleri değil, sonrasında da takibini yapmaya çalışıyoruz projelerin. Fikri haklar konusunda da yine kısaca size bilgi vereceğim. Süreçlerimizle ilgili yeniliklerden bahsedeceğim. Ana hatlarıyla sunumumuz bu şekilde olacak. 2018-2022 yılları arasında TÜBİTAK, ARDEP toplamda 52.331 tane proje başvurusu sağladı. Bunlardan 8.307'sini destekledi ve 18 milyar 582 milyonun üzerinde bir destek bütçesi ve şu ana kadar bunun 17 milyar 921 milyonu aktarılmış durumda. Hızlıca ARDEP'teki programlara bakmak gerekirse 1002 hızlı destek programımız. Bunu yakın tarihte ikiye böldük. 1002A ve 1002B şeklinde. 1002A projeleri 12 ay 60 bin liraya kadar desteğimiz var. 1002B ise acil destek modülü, devam eden bir projeyi destekleme amaçlı ya da devam eden bir yüksek lisans çalışması, doktora çalışmasını tamamlama amacıyla sunulabilecek proje tiplerimiz. Burada en büyük avantaj doktora öğrencilerine yürütücü olma fırsatı tanınıyor ve de diğer avantajı da şu: kadrolu personel olmasına ihtiyacı yok doktora öğrencilerinin bunu sunarken. Bu program kapsamında biz 2008 ve 2022 yılları arasında 29.562 proje önerisi aldık ve bunlardan 6.261 tanesini destekleyerek toplamda 1.14 milyar liralık bir bütçe aktarımını gerçekleştirmiş olduk. 2022 yılında bu uygulamaya başladığımız 2022 yılı itibariyle de 44 doktor öğrencimiz buradan proje üyesi olarak destek aldılar. 1005 programı benim için özel de bir program. İşe ilk başladığım zaman ortaya çıkan bir programdı ve uzmanlık tezimde 1005 programının iyileştirilmesi üzerine yapmıştım. 1005 programının asıl amacı dışa bağımlı ürün veya teknolojiye yeni özellikler kazandırarak yerli üretimi amaçlayan projelerin geliştirilmesi ve bu program kapsamında yaygın bir teknoloji, sistem veya sürece yeni özellikler kazandırarak yeni malzeme, ürün, süreç ve hizmet geliştirilecek projelerin desteklenmesi. 2017-2022 yılları arasında 1005 programı kapsamında 16 tane patent ve tescil alınmış durumda. Yine bu programın ilk başladığı gün itibariyle 2013'ten 2022'ye kadar 2576 proje önerisi değerlendirmeye alınmış ve bunlardan 295 tanesi desteklenerek 314.6 milyonluk bir bütçe aktarımı sağlanmış. 1003 programı bizim için önemli bir program. Öncelikli alanlar, Ar-Ge projenin destekleme programı, çağrılı bir program. Uzun zamandır çağrıya çıkmadık ancak TAGEM'le yaptığımız protokol kapsamında yakın tarihte bununla ilgili bir çağrıya çıktık. Bu çağrı öncesinde biz 10 yıllık süreçte 9.857 proje önerisini aldık ve bunlardan 551 tanesini destekledik ve 3.36 milyarlık bir bütçe aktarımı gerçekleştirdik. Grafikten de görebiliyorsunuz, özellikle son yıllarda bu konuda çağrıya çıkmadığımız için grafik bu şekilde ilerlemekte ama yeni çağrımız kapsamında da yine bayağı desteklerimiz proje söz konusu. 1003 sadece TOVAK özelinde, benim çalıştığım birim özelinde değil, bütün ARDP özelinde farklı alanlarda, farklı öncelikli alanlarda destek sağlayan bir program. Burada gıda toplam ayrılan bütçenin %10'unu kapsamakta, bu öncelikli alanlar kapsamında aktardığımız bütçelerde. Biraz önce bahsettiğim TAGEM'le yaptığımız ortak çağrı, bu sadece 1003 kapsamında değil, 1003 ve 1005 programları kapsamında yapmıştık. Ancak özellikle bugünkü konumuz tarımsal biyoteknoloji olduğu için ve 1005'te de tarımsal biyoteknoloji alanında proje gelmediği için ben size biraz daha tarım çağrısı üzerinde 1003'ten bahsetmek istiyorum. Bu yeni tamamladığımız bir çağrı. Biz özellikle öncelikli alanlarımızı belirlerken TAGEM kendi öncelikli alanlarını belirledi. TÜBİTAK'ın politikalar birimi kendi öncelikli alanlarını belirledi ve özellikle ıslah çalışmalarında hep aynı ifadeleri kullanmaya çalıştık. Yeni biyoteknolojik yöntemlerin kullanılması, markör geliştirmesi, CRISPR-Cas teknolojilerinin kullanılması ya da biyotik ve abiyotik streslere dayanıklı tolerant hatların ortaya konmasında yeni biyoteknolojik yöntemlerin kullanımı ve özellikle biraz önce Kemal Hocam'ın da bahsettiği markör geliştirme ve CRISPR-Cas teknolojilerinin kullanılmasını biz teşvik etmeye çalışıyoruz. Bu tarz projeleri önceliklendirmeye çalışıyoruz ve diğer programlarımızda da bu tarz projeleri, bu tarz teknikleri kullananlara ek puan verme yoluna gidiyoruz teşvik etme amacıyla. Genel olarak tarım 1003 çağrısına baktığımız durumda 186 adet 1003 projesi başvurusu almış durumdayız. Bunlardan 28'i kabul edildi. Yani yaklaşık %15'lik bir destek oranı söz konusu ve bunların sözleşme süreçlerini başlattık. Bu 28 projenin 9'u tarımsal biyoteknoloji kapsamında değerlendirilebilecek projeler. Yani toplam kabul edilen projelerin %32 aslında tarımsal biyoteknoloji ile ilgili. Neredeyse 1/3 diyebileceğimiz bir oran. 31 proje için geliştirilebilir kararı verildi. Yani reddedildi ama tekrar bir opsiyon sunuldu. Bu hocalarımız değişiklikleri yaptılar, bize tekrar sundular. Bu 31 projenin şu anda değerlendirme süreci devam ediyor. En yakın zamanda bunlarla ilgili sonuçları aktaracağız. Ve bunların içinde de yine 7 tanesi tarımsal biyoteknoloji alanında kapsamında değerlendirilebilecek projeler. Bir diğer programımız 3501 kariyer geliştirme programı. 36 ay destek süresi ve 600 bin liraya kadar destek verebiliyoruz. Yılın her günü başvuru yapılabiliyor. Bu program kapsamında yaptığımız değişiklikte biz yürütücülerimizi de artık proje değerlendirme süreçlerine dahil ediyoruz. Panelistlerimiz proje değerlendirdikten sonra yüz yüze bir şekilde soru cevap kısmına geçiyoruz. Proje yürütücüsü adayı hocalarımız panelistlerle yüz yüze soru cevap kısmını gerçekleştiriyorlar. Bunun haricinde eskiden daha önceleri doçentlere bu programa başvurma imkanı verilmiyordu ama artık bu imkan da veriliyor. Mümkün olduğu kadar süreçlerimizi hep değiştirmeye, geliştirmeye çalışıyoruz. Kariyer geliştirme programı kapsamında 2008-2022 yılları arasında 8.392 proje önerisi alınmış. Bunlardan 1731 tanesi desteklenerek 2.38 milyar liralık bir bütçe aktarımı gerçekleştirilmiş. Ve 1001 programı bizim amiral gemimiz. 1965'ten beri destek verdiğimiz bir programımız. En fazla başvuru olan program. Toplamda 1.250.000 liraya kadar bir destek miktarımız var ve 36 aya kadar destek verebiliyoruz. 2008-2022 yılları arasında 54.000 proje başvurusu alınmış ve bunlardan 8.983 tanesi desteklenerek 17.25 milyar liralık bir bütçe aktarımı gerçekleştirilmiş durumda. Biraz önce bahsettiğim TAGEM'le beraber çıktığımız 1003 çağrımız vardı. Bu çağrı aslında sadece yani işbirliğimiz sadece bu çağrı kapsamında sınırlı değil. TAGEM'le yaptığımız bir protokol var ve biz diğer programlarımızdaki öncelikli alanları belirlerken yine TAGEM'le işbirliğine gidiyoruz. Şu anda ekranda gördüğünüz 2021 yılındaki 1001 programında ek puan verme hususlarındaki kriterlerimiz. Eğer bu alanlardan projeler gelirse biz bu projelere 1001 projelerindeki puanlarına ek olarak puan verebilme durumumuz söz konusuydu. Bu sene itibariyle bunu biraz daha güncelledik, biraz daha geliştirdik ve hatta tarımsal biyoteknoloji alanına daha da iyileştirdik diyebiliriz. Özellikle sağ tarafa bakarsanız sağ tarafta yeni nesil DNA, RNA dizileme teknolojinin kullanımı, CRISPR-Cas teknolojisinin kullanımı, hızlı speed breeding imkanlarının kullanımı, doku kültürüyle klasik yollar çoğaltılması güç olan bitkilerin in vitro mikro çoğaltımı, klasik ve biyoteknolojik yöntemler kullanarak iklim değişikliği sonucu olacak biyotik ve abiyotik stres koşullarına toleransa dayanıklı bitkilerin geliştirilmesi, virüsten hariç bitkilerin moleküler markörleriyle seçilimi ve çoğaltımı gibi daha da çoğaltabileceğimiz başlıklar söz konusu. Tarımsal biyoteknoloji anlamında bu alanda gelen projelerimize biz bu 1001 dönemlerinde de ek puan uygulamasına uyguluyoruz. Özellikle yeni nesil teknolojilerin kullanılmasını teşvik etmeye çalışıyoruz. Bir diğer programımız 1071 Uluslararası Araştırma Fonlarından yararlanma kapasitesinin ve uluslararası Ar-Ge işbirliklerinin katılımını arttırması yönelik destek programı. TÜBİTAK'ın ikili anlaşmalar kapsamında 65 ülkeden 93 farklı kurum ile işbirliği mevcuttur. Çok taraflı platformlar aracılığıyla da 100'den fazla ülkeyle işbirliği yapabilmekteyiz. 2022 yılında 28 ikili işbirliği ve 23 çoklu işbirliği çağrısı yayınlanmış durumda. 1071 programımızın yapısı biraz değişik. Sadece ARDEP yani akademiye yönelik bir çağrı değil, TEDEP benzeri özellikler de taşımakta. Farklı tematik alanlarda uluslararası çağrılara çıkılabilmekte. Üniversite, sanayi, kamu işbirlikleri desteklenmekte. Çağrı üzerine destek miktarı ve süresi belirlenmekte. Benim bir önce bahsettiğim gibi ARDEP'in 1001 ve TEDEP'in 1509 programlarının bir arada harmanlayan bir program olduğunu söyleyebilirim. Uluslararası programlar kapsamında da yine ekranda gördüğünüz şekilde 2008-2022 yılları arasında 11.753 proje önerisi alınmış, 2.192 proje önerisi desteklenerek 4.17 milyarlık bir bütçe aktarımı gerçekleştirilmiş. Genel olarak baktığımız zaman, program ayırt etmeksizin baktığımız zaman 2002 yılında TÜBİTAK ARDEP'e 1198 projesi önerilmişken bu sayı 2022'de 10.928'e çıkmış. 2002 yılında yürürlükteki projeleri 113.4 milyon lira aktarırken bu tutar 2022'de 3.55 milyar liraya ulaşmış. Tabii ki bu tutarlar hep deflate tutarlar. Yani aslında o zaman aktardığımız tutar 113.4 milyon değil. Enflasyon oranında arttırdığımız zaman 113.4 milyon tutarı hesaplanmış durumda ve bu açıdan karşılaştığımız zaman da çok ciddi bir fon aktarımı sağlamış durumdayız. Araştırmacılarımızın bu sunuma katılanların büyük bir kısmı ARDEP'in az çok yapısını biliyorlardır. Ama hani çok kısaca hatırlatmak gerekirse TOVAK, Tarımsal Araştırmalar, Tarım, Orman, Veteriner, Gıda Araştırma Destek Grubu ve toplam ARDEP içinde bizim 2012-2022 yıllarında verdiğimiz desteklerin fon açısından oranı %4.38. Ancak KAMAK, kamu araştırmalarının oranının yüksek olduğunu görüyoruz. %27.47. Burada da yine ziraatla ilgili araştırmalara destek verilebilmekte. Ana hatlarıyla baktığımız zaman 2022 yılındaki destek oranlarımızı ARDEP için %24 olarak verdik ama farklı programlara farklı destek oranlarını görebilmekteyiz. 3501 proje için %24, 1002 proje için %25, 1001 için %21, 1005 için %19 ve uluslararası proje için %34'lük bir kabul oranımız mevcut. Aslında baktığımız zaman şunu söyleyebiliriz. ARDEP'e bir proje sunduğu zaman 1/4 ihtimalle kabul edilme şansınız mevcut. Buraya gelmeden önce biraz daha derinlemesine özellikle bu oranları incelemek istedim. Hani hem ARDEP özelinde bir alt kırılıma indiğimiz zaman çalıştığım grup olan tarım grubundaki oranları size sunmak istedim. Hem de bugünün konusu tarımsal biyoteknoloji, özellikle yeni nesil biyoteknoloji teknikleriyle ilgili konulardaki destek oranlarımızda sizlere bunları sunmak istedim. Burada özellikle son 3 yılın verilerini verdim. Bu 3 yılın özellikleri de var. 2022 yılı aslında yani geçtiğimiz yıl bizim normalleştiğimiz yıldı. 2020 biliyorsunuz pandemi yaşadık, kapanmalar yaşadık ve o dönem tek dönem çağrıya çıkabildik 1001 için. 2021'de de bizim için çok ekstra yoğun bir dönemdi. TAGEM'le yaptığımız protokol ilk defa bu dönem yürürlüğe girdi ve orada da bir TOVAK özelinde proje patlaması yaşandı. Normalde beklediğimizin 2 katı kadar proje geldi. Genel olarak oranlara bakmamız gerekirse 2020 yılında ARDEP'e 1183 proje geliyor ve bunlardan 213'ü destek veriyor ve destek oranımız %18. TOVAK'a gelen proje sayısı 277 ve tek dönem çıktığımız için 39 proje destekleniyor ve destek oranımız %14. Bu dönem, bu yıl aslında belki de tarımsal biyoteknoloji anlamında en kötü yılımızdı. 26 tarımsal biyoteknoloji projesinden sadece 2 tanesi desteklendi. Bir sonraki yıl yine dediğim gibi TAGEM'le yaptığımız protokol kapsamında proje sayısında müthiş bir artış gördük. Bu bizi biraz yordu ama bir açıdan da mutlu etti. Yani rağbet görmek, bizim yorulmamız iyi bir şey aslında. Bu akademisyenlerin çalıştığını gösterir. 753 proje geldi TOVAK'a ve bunlardan 87'si desteklendi. %11.50 gibi bir destek oranı söz konusuydu. Tarımsal biyoteknoloji projenin sayısı ise bu 753 proje içinde 72 ve 9 tanesi desteklendi. %12.5 gibi bir destek oranı söz konusu tarımsal biyoteknoloji projenin kendi içindeki oranı. Demin de bahsettiğim gibi 2022 bizim normalleştiğimiz yıldı. 2 dönem çağrıya çıktık ve TOVAK'a 510 proje geldi. Bunlardan 84'ü desteklendi ve TOVAK'ın ideal destek oranı %16 gibi bir orana ulaşmış olduk. Ama tarımsal biyoteknolojide ise bir artış gözlemledik. 54 projenin 11'i desteklendi. Yani %20 gibi bir oranı yakaladık. Diğer TOVAK'ın altındaki diğer alanları destek oranı anlamında biraz daha geçmiş olduk. genel itibariyle şekli son 3 yıldaki tarımsal biyoteknolojideki değişimi en azından bu istatistikleri size sunmak istedim. Peki deminden beri bahsediyoruz. Bu tarımsal biyoteknoloji projeleri neleri kapsıyor? Yine kapsamlı bir çalışma yapmaya çalıştım. 2001-2023 yılları arasında TÜBİTAK'ta desteklenen tarımsal biyoteknoloji projeleri. Toplam desteklenen proje sayımız bu 23 yıllık, 22 yıllık periyotta 4347 ve bunlardan 618 tanesi tarımsal biyoteknoloji alanında sayılabilecek projeler. Bir şekilde gruplandırmaya çalıştım. Markör çalışmaları, Genome-Wide Association Studies, QTL Studies, SMP çalışmaları. Burada toplamda 187 tane projemiz desteklenmiş durumda. Gen karakterizasyonu, özellikle PETFAY ya da yolak mekanizma çözümleme çalışmalarında 100 projemiz desteklenmiş. Bitki hastalıkları, bitki koruma alanındaki özellikle moleküler çalışma ve tarımsal biyoteknoloji diyebileceğimiz alanda da direnç genleri, patojen analizleri, bitki patojen ilişkilerine dair projelerde yine geldi grubumuza. Bunlardan da 110 tanesi desteklenmiş durumda. Bitki doku kültürü çalışmaları, micropropagation, cryopreservation, secondary metabolite production ile ilgili 83 projemiz desteklenmiş durumda. Omik teknoloji diye gruplandırdık. Yeni nesil sekanslama, RNA sekanslama, transkriptom analizleri, genom sekanslama, proteom, mikroRNA sekanslama alanında örnek verebileceğimiz omik teknolojilerine de 65 tane projemiz desteklendi. Toprak, bitki ve mikroorganizma ilişkileri. Bunu daha çok toprak ve bitki besleme diye de olarak Ziraat Fakültesi altında değerlendirebiliriz ama burada biraz da ilişkiler devreye giriyor. Örnek teknolojilerde şu şekilde söyleyebiliriz. Fitoremediasyon, plant growth promoting bacteria, fungi, rhizobacteria and plant interaction, nanotech and fertilizers şeklinde gruplandırabileceğimiz alanlarda 50 projemizde desteklendi. Ve en sonunda belki de bugünün en hot topiklerinden biri genom editing'de. Burada da 23 adet projemiz desteklenmiş durumda. Burada hangi teknolojiler var? CRISPR teknolojileri var. Viral Induced Gene Silencing teknolojileri var. RNAi teknolojileri var ve burada da yine 23 tane projemiz desteklenmiş durumda. Oranlara bakacak olursak özellikle TÜBİTAK'ta desteklenen tarımsal biyoteknoloji projelerinde markör çalışmaları büyük bir ağırlığı var. %30 gibi bir oranımız var 187 projeyle. Bunun ardından gen karakterizasyonu, bitki hastalık çalışmaları gelmekte. Genom düzenleme çalışmaları yeni yaygınlaşmaya başlamakla birlikte daha önceki yıllarda 2010'lu yılların başında daha çok RNAi teknolojisi, Viral Induced Gene Silencing gibi düşünebileceğimiz teknoloji yavaş yavaş artık kendini CRISPR ve CRISPR'ın türevlerine bırakmış durumda. Şu ana kadar 23 proje destekledik ama emin olun çok fazla hem 1001 kapsamında hem 1003 kapsamında çok fazla CRISPR projesi geliyor ve şu anda zaten devam eden bir 1001 dönemimiz var. Umarım olumlu sonuçları da araştırmacılarımıza bununla ilgili vermeyi istiyoruz. Daha fazla CRISPR projesi desteklemekte istiyoruz. Biraz önce size aslında programlarımızdan bahsettik ve bu tarımsal biyoteknoloji projelerinde gruplandırmaya çalışmıştım. Bunları bir de programlara dağılımını da sizlere göstermek istiyorum. Evet, bahsetmiştim. 1001 bizim amiral gemimiz. Daha çok araştırmacılarımız, yürütücülerimiz hep 1001'i tercih ediyorlar. Ve toplamda da tarımsal biyoteknoloji projenin 618'in 287'si neredeyse yarısı 1001 projesi. Ve her başlık içinde aslında hep en büyük oran 1001'de. Yani 23 tane genom düzenleme projesi desteklemişiz. Bunlardan 9 tanesi bu kapsamda. Uluslararası projeler de aslında 1001 esaslarına tabi. Onu da aslında 1001 gibi değerlendirebiliriz. Ama burada özellikle marker çalışmaları biz 2013-2014 yıllarında ziraat alanında, tarla bitkilerinde çok fazla çağrıya çıktık. Bu alanda özellikle marköre dayalı seleksiyon, MAS alanında çok fazla projede destekledik. 1003 kapsamında da en fazla marköre dayalı seleksiyon çalışmalarını desteklemiş durumdayız. Sunum biraz hızlı gidiyor. Hem programa da biraz yetişmek anlamında hızlı gidiyorum. Umarım sıkıntılı bir süreç yoktur. Sonrasında soru cevabı zamanımız kalırsa yine cevaplayabilirim. Çok kısaca Çanakkale 18 Mart Üniversitesi'nin de durumunu biraz daha ortaya koymak istiyorum. 2013, 2012, 2022 yıl arasında ARDB bu üniversiteden 1163 proje önermiş ve 212 projenin destek kararı verilmiş durumda. Son döneme doğru son 5 yıl program bazlı baktığımız zaman yine üniversitemize daha çok 1001 programını tercih ediyor. 562 proje verilmiş ve 89 projenin desteklenmesine karar verilmiş. Bu konuda şanslıyız. En çok TÜBİTAK tercih ediliyor. Çanakkale 18 Mart Üniversitesi'nden TÜBİTAK'a çok fazla proje geliyor ve biz de mümkün olduğu kadar destekliyoruz bunları. Son 10 yılda aktarılan destek miktarlarını da yine ekranda görebiliyorsunuz. 219.57 milyon liralık bir destek sağlanmış durumda toplamda Çanakkale 18 Mart Üniversitesi'ne. Destek oranlarında, genel destek oranını düşündüğümüz zaman Türkiye ortalamasının üzerinde bir oran, özür dilerim, çok az da olsa altında bir oran söz konusu 18 Mart Üniversitesi için. Mümkün olduğu kadar biz panelist seçerken yaygın dağılıma dikkat ediyoruz ve Çanakkale 18 Mart Üniversitesi'nde çok kıymetli akademisyenlerimizi panelist olarak panellerimize davet ediyoruz. Deminki istatistiklerin devamı niteliğinde yine bunu söyleyebilirim. Tarımsal biyoteknoloji projelerinde Çanakkale 18 Mart Üniversitesi'nin durumu nedir? Burada da yine ekranda gördüğünüz şekilde büyük bir çoğunluğu yine 1001 projesi, 1001 programı kapsamında sunulan projeler var. Marka dayalı çalışmalar söz konusu. Bitki koruma ile ilgili projelerimiz de var. Özellikle yine toprak, bitki ve mikroorganizma ilişkilerine dair yine projelerimiz de bu üniversite kapsamında bize sunulmuş ve desteklenmiş durumda. Destek haritası Türkiye'nin geneline herkese ulaşmaya çalışıyoruz TÜBİTAK olarak. Tabii ki büyük şehirler çok fazla sayıda da üniversite olması ve kapasitenin fazla olması bile onlara daha fazla para aktarıyoruz ama mümkün olduğu kadar her yere ulaşmaya elimizin değmesine çalışıyoruz. Yine mümkün olduğu kadar her üniversiteden panelist çağırmaya çalışıyoruz ama tabii burada İstanbul, Ankara, İzmir biraz daha fark yaratabiliyor üniversite sayısının ve kapasitelerinin fazla olmasından dolayı. Buraya kadar genel olarak hani, tarımsal biyoteknolojiye dair desteklerimizi ve programlarımızı özetlemeye çalıştım. Bizim en büyük derdimiz projeler bittiği zaman sonrasında ne olacağı. Yani bunların takibinin yapılması. Süreçlerimizle ilgili bununla ilgili geliştirmeler de yapıyoruz. Biz artık her gelişme raporu, her sonuç raporuyla beraber fikri ürün bildirim formunun doldurulmasını istiyoruz ve bunun takibini yapmaya çalışıyoruz. Eğer bir tescile başvurusu olacaksa muhakkak bilgilendirilmek istiyoruz. Patent başvurusu yapılacaksa süreçleri takip etmek istiyoruz ve bununla ilgili de biz yürütücülerimizi her şekilde bilgilendiriyoruz. Her şekilde de yardıma hazır olduğumuzu belirtiyoruz. Biraz önce bahsettiğim programlarımızın yenilenmesi için sürekli değişiklikler, sürekli iyileştirmeler yapıyoruz. Başkanımız da toplantılara katılıyor, şikayetlerinizi dinliyor ve onun ardından da bize ne konuda çalışma yapmamız gerektiğini bildiriyor. Bununla ilgili yapılan son yenilikleri de burada çok kısaca tek slaytta özetlemek istiyorum. Bazı durumlarda tekrar sunulan projelerde yürütücülerimiz kendilerini yeterince ifade edemeyebilir yazılı olarak diye video yükleme imkanı veriliyor. Başvuru aşamasındaki bürokratik işlemleri, evrak kalabalığını azaltmak için başvuru aşamasında özellikle izin belgeleri, etik kurul belgeleri istemiyoruz. Bunlar proje desteklendiği zaman tamamlatılan belgeler, hatta proformalar da aynı şekilde. Biz açık bilim politikası kapsamında birazdan bahsedeceğim. Veri yönetim planı istiyoruz. Elde edilecek verilerin nasıl paylaşılacağı, hangi platformlarda paylaşılacağı konusunda bilgi istiyoruz. Süreçlerimizi e-imza süreciyle hızlandırdık. İlk defa başvuru yapacak deneyimsiz hocalarımıza, deneyimli hocalarımızın yanında ezilmemesi için pozitif ayrımcılıkla beraber ek puan uygulaması yapıyoruz. Malumunuz asgari ücretin artmasıyla beraber biz burs limitlerini her seferinde arttırıyoruz. 3501 ve 1005 programlarında proje başvuru mali kısımlarda üst limitlerimizi arttırdık. 1002 programımızı ikiye böldük. 1002A ve 1002B şeklinde sunumun başında yine bundan bahsetmiştim size. 1001 programımıza yine limitlerimizi arttırdık. Ve artık projeler kapsamında, ARDE projeleri kapsamında desteklenen proje yayın desteği imkanı sağladı. Belki de en büyük sıkıntılardan biridir. Yayın için belli bir ödenek gerekiyor. Bunu artık çıktıların yayılımı kapsamında da bizden, TÜBİTAK'tan talep edebileceksiniz. Burada yine yeniliklere dair detaylarımızı da görebiliyorsunuz. Burs limitlerine dair iyileştirmelerimiz, hatta son zamanlarda yine bir iyileştirme yaptık. Sigorta konusunda doktor öğrencilerine isteğe bağlı sigorta yaptığı zaman ekstra yine para verilmeye durumu söz konusu. 1001 programı kapsamında özellikle Avrupa Birliği programlarına başvuru yapmasını istiyoruz hocalarımızın. Avrupa Birliği'nde Ufuk 2020 programına başvuru yapmış hocalarımıza ek puan uygulamamız var. İşbirliklerini önemsiyoruz. Projeler arasında işbirliği, çoklu disiplin arası projenin olmasını istiyoruz. Eğer projeler bu şekilde gelirse onlara yine ek puan veriyoruz. Yeşil mutabakata uyum konusunda yol haritamız var TÜBİTAK olarak. Projeler eğer yeşil mutabakata uyumlu olduğunu bildirirlerse ve panelistler bu konuda hemfikir olurlarsa burada da yine ek puan uygulamamız söz konusu. Ve size yine sunumun başında belirttiğim özellikle tarım alanında TAGEM'le yaptığımız öncelikli alanlar belirleme kapsamında oradaki başlıklarla uyum olması durumları, yani TÜBİTAK'ın ve TAGEM'in öncelikli alanlarında da proje sunduğunuz durumda yine ek puan verebiliyoruz 1001 programı kapsamında. Burada da biraz önce bahsettiğim platformumuzu biraz anlatacağım sizlere. Aperta, TÜBİTAK'ın açık arşivi. Burada nelere ulaşabileceksiniz? Sayısal veri, dokümanlar, lab notları, anketler, cevaplar, kopyalar. Burada amacımız nedir? Hızlı, kaliteli ve harcamaları minimuma düşürmek. Hızlı bir şekilde veriye ulaşmak, kaliteli veriye ulaşmak ve harcamaları düşürmek. Doğrulanabilir, hatasız, tekrarlanabilir, eksiksiz veriyi herkese ulaştırma amacıyla oluşturuldu bu platform. Burada eğitimler de söz konusu. Bu platformumuzdan herkes faydalanabilir. Sağ alt tarafta da adresini görüyorsunuz, Aparta'nın. Ben bu noktada da çok kısaca, ben yaklaşık 10 yıldır tarımsal biyoteknoloji projenin değerlendirme süreçlerinde sekreteryasını gerçekleştiriyorum. Ama artık biraz öğrendim. Bu konuda da sizlere çok kısaca belki birkaç uyarı ya da hatırlatma gibi bilgi vermek istiyorum. Özellikle tarımsal biyoteknoloji projelerinde evet temel bilimler çok önde. Çok üst düzey yeni teknolojiler kullanılıyor ama bunların tarımsal çıktıları ya da ekonomik faydası konusunda proje yürütücülerimiz yeterince önem vermiyor. Bunları belki de açıklama tercih etmiyorlar. Bu temel bilimler projesi gibi düşünüyorlar ama yani tarımı yapılan bir bitki üzerinde bitki biyoteknolojisi çalışması yapıyorsanız bunun muhakkak ileriki safhalarda, yakın vadeli, kısa vadeli olmayabilir ama uzun vadeli tarımsal çıktıları ve ekonomik fayda konusunda bir iki satırda olsa bir şey yazmanızı sizlere tavsiye ediyorum proje başvurusu aşamasında. GDO için yasal düzenlemeler ve CRISPR'a dair yasal düzenlemeler ne olacağı net değil. CRISPR'a dair çok olumlu haberlerin geleceğine dair dedikodular geliyor kulağımıza. Bilemiyoruz ne olacak ama buna dair de genelde panelistlerimiz projeleri İster istemez eleştiriyor. Evet, CRISPR'la bu yapılabilir, bu değişiklik yapılır. Müthiş bir teknoloji, altyapımız var, uygulanabilirdi ama bunu yaptıktan sonrasına dair bir şeyleri öngöremiyoruz bu yasal düzenlemelerden dolayı. Belki ileriki günlerde bu yasal düzenlemelerle ilgili değişiklikler olursa hem GDO için hem CRISPR için daha farklı bir açılımda gerçekleştirilebilir. Bunlar hep genelde yaygın etkiye, çıktılara, etkiye yönelik tespitlerim ya da önemli noktalar, değinmek istediğim noktalar. Bir diğer husus da Türkiye'de biyoteknoloji konusunda çalışan çok fazla araştırmacı var. Hepsi de çok kaliteli araştırmacılar. Çok ciddi çalışmalar gerçekleştiriliyor. Altyapımız iyi. Bir nokta hariç özellikle belki de yeni nesil sekanslama ile ilgili ciddi bir sıkıntımız var. Herkes hizmet alımı yolunu tercih ediyor. Evet olabilir ve yurt dışını tercih ediyor. Buna dair aslında herkesin de bir soru işareti var. Yani neden yurt dışına gönderiyoruz? Türkiye'de neden bu altyapıları oluşturamıyoruz? Özellikle İzmir'de İBG, Kocaeli'de, Gebze'de MAM, bu işleri aslında yapabilen kurumlarımız, yeni nesil sekanslama ve hizmet alımları konusunda bu konuda proje verecek hocalarımız için en azından akıllarında olsun bu tarz merkezler. Hatta Ankara'da da yeni buna benzer bir enstitünün açılması ya da merkezin açılması da gündemde. Yeni nesil sekanslama ve hizmet alımları konusunda da öncelikli olarak bizim tabii ki böyle bir sınırlamamız ya da yorum, yönlendirmemiz olmaz ama ben sadece aklınızda bulunsun diye bunu da hatırlatmak istiyorum. Sosyal medya kanallarından bizi her türlü takip edebilirsiniz. Ben tekrar burada mail adresimi de vermek istiyorum. serkan.tuna@tubitak.gov.tr sunum burada tamamlıyorum. Tekrar teşekkür ediyorum. Hem ben önceki Kemal Hocama çok teşekkür ediyorum sunum için hem de bundan sonraki çok kıymetli profesörleri yapacağı sunumdan dolayı teşekkür ediyorum. Mümkün olduğu kadar onların zamanından çalmamak için hızlı gittim. Umarım anlaşılabilir olmuşumdur. Hepinize çok teşekkür ederim. Keyifli bir etkinlik diliyorum.
Thank you, Serkan Tuna, for your informative presentation. Now we will move on to a brief Q&A session where I will present the questions to you. I have a few questions for you. Hocam sizin için birkaç adet sorum var. İlk sorumuz CRISPR-Cas9 gibi teknolojileri öğrenebilmek ve bu alanlarda eğitim alabilmek için lisans ve yüksek lisans öğrencileri adına yurt dışı olanakları ve burs imkanları var mıdır?
BİDEB'in özellikle lisansüstü öğrencileri için dönemlik bursları söz konusu. Bunun haricinde özellikle araştırmacılar, Türkiye'deki yerleşik araştırmacılar BİDEB'e etkinlik, organizasyon burslarına başvurabiliyorlar. Hatta çok yakın tarihte CRISPR ile ilgili bir etkinlikte gerçekleştirildi. Ama bunlar genelde butik oluyor çünkü workshop şeklinde oluyor. 10 kişiyle sınırlı oluyor, 20 kişiyle sınırlı çünkü uygulama da yapılabildiği için ama yani öğrenciler bir araya gelip bir hoca da bulup özellikle BİDEB'e, ARDEP'in böyle bir şey yok ama BİDEB'e, Bilim İnsanı Destekleme Başkanlığı'na başvurabilir. Etkinlik organizasyonu için desteklerimiz de mevcut.
İkinci sorumuz Fatih hocamızdan geliyor. TÜBİTAK'ın genom editing bitkileri hakkında Türkiye regülasyonunun bir çalışması var mıdır?
Çok özür dilerim. Tekrar alabilir miyim soruyu?
TÜBİTAK'ın genom editing bitkileri hakkında Türkiye regülasyonu hakkında bir çalışması var mıdır?
TÜBİTAK genelde regülasyonlar konusunda çok dahil olmuyor. Bunlarda daha çok bakanlık devreye giriyor. TÜBİTAK sadece uygulayıcı ya da zaten TÜBİTAK'ın asıl amacı araştırmaya destek verdiği için araştırmaya dair de herhangi bir şu anda sıkıntı olmadığı için bir çalışmamız bu konuda yok.
Pekala hocam, çok teşekkür ediyorum. Çok sağ olun. Sorularımız
teşekkür ederim.
vardı. Çok teşekkür ediyoruz. Before our next presentation, I would greatly appreciate that if your speakers could kindly speak at a slightly slower pace, taking into consideration that the needs of the interparents. Thank you. And now our next speaker is Professor Dr. Luigi Cativelli, the Director of the Research Center for Genomics and Bioinformatics, and at the Council for Agricultural Research and Economics in Italy. He will be presenting on new genomic techniques for plant breeding in Italy, main results of the National Biotech Project. Feel free to ask questions in the chat section during the presentation, and the selected questions will be addressed during the Q&A session. Please proceed, Professor. Welcome.
Okay, thank you very much for this introduction. It's an honor and a pleasure to be here with all of you to present what has been the experience in Italy during the last 5-6 years working on new genomic techniques. I also hope to be interpreter friendly enough, don't speak too fast. My presentation is split in 3 parts. The first part is dedicated to an introduction to the system, to the genome editing system. The second part, which is the largest, I will tell you about the experience of this biotech project in Italy. And the final part of the presentation, I will talk about some regulation issue, at least some regulation issue from an European point of view. Okay, so as a starting point, I will try to make this overview of the breeding effort, simply to put the new genomic technique in the context of plant breeding. Everybody of us know that plant breeding is based on biodiversity. So if you work with specific crop, you actually work with a gene pool of your crop, and the gene pool is made of all genes present in cultivated, wild relative, land races, and whatever. However, out of a very large gene pool, what is currently used in breeding is a small pool of genes. And usually we have this definition, elite cultivar, because there are few cultivars that are currently used in agriculture, and this cultivar represent usually the best combination among the best genes or alleles. And what a breeder does day by day is simply to improve this best combination. And from time to time, you also introduce new allele or new genes in the elite cultivars. This introduction is fundamental if you want to face, for instance, new diseases. And to introduce this new allele, the standard approach is to screen the gene pool, find an interesting gene or allele, and then make an introgression into your elite cultivars. In principle, it can be represented in this way. You screen, and then you find the gene of interest, and you transfer the gene of interest in the elite cultivar. This transferring can be done using traditional backcross program, backcross implemented marker-assisted selection, or you could also take the gene from your wild and transfer the gene directly in the elite cultivar using transformation. This goes under the definition of cisgenesis, which is a kind of transgenesis that is particularly of interest in Europe, and I will present you some more details later. But there are situations where specific traits cannot be found in the gene pool. You need a new kind of gene, a new allele. So you can produce a new allele, and as a standard in the past, people were used to generate new allele using mutagenesis. Genome editing is a system that can replace with much higher efficiency the traditional mutagenesis. So genome editing and cisgenesis goes under the umbrella of new genomic techniques, and at least in Europe are seen as an alternative to traditional breeding process. There are also extreme situations where traits cannot be found in the gene pool of your cultivated crop, cannot be easily generated using mutagenesis or genome editing, then if this is the situation, the only solution is to take a trait for another organism, another living organism, make a GMO plants, introduce your trait into the elite cultivar of your crop. This is a GMO. This, at least in Europe, GMO are really, the public opinion is generally against GMO, and there is strong regulation which limit the application of GMO in European agriculture. But what we can see in this perspective is that the new genomic technique offers two shortcuts for improving elite cultivars. Probably the most relevant technology is this called genome editing. So genome editing is, well, there are several tools to make genome editing, but the most widely used, the only one probably used, is this CRISP, known as CRISP-Cas9. CRISP-Cas9 is a biological mechanism developed by bacteria as a defense system against viruses. And instead, this system is simply capable to recognize and cut specific DNA sequence. I will spend a few minutes to tell you a bit more about the history of this system, because to my understanding, it's really a fantastic story. This is a biological system naturally evolving bacteria and based on two compounds. One is called CRISP, another one is called Cas9. CRISP is an abbreviation that stands for clustered regularly interspersed short palindromic repeats. CRISP is a region of the bacteria genome. It is a region of the bacterial genome where the bacteria, as evolutionary speaking, accumulated short sequence deriving from viruses. This sort of catalog of short sequence corresponding to different viruses' DNA. Cas stands for CRISP-associated protein. And indeed, in the bacterial genome, there are many CRISP-associated proteins. The Cas number 9 is the one that is the active in the recognition and in the cutting of the DNA target. In the next slide, I have a video. I will introduce this video. This is a video from the Max Planck Foundation, and is a cartoon that explains very easily how the system works. We can split the video in 4 parts. The first part represents the evolutionary mechanism that allow the bacteria to accumulate specific virus DNA sequence in the CRISP region. Let's go in together. Okay, as a standard, when a virus infects a bacteria, there is simply inject its own DNA inside of the bacterial cell. That is a standard infection process for any virus. So here you see the virus, and the virus is now injecting the DNA within the bacterial cell. This is the virus DNA, the one in red. And under specific condition, the bacteria is able to cut the virus DNA, to cut a fragment of virus DNA and integrate this fragment in the CRISP region of the bacterial genome. This is an evolutionary story, so not every infection works in this way, but there are specific situations where this is possible. Once the virus has integrated the fragment of, when the bacteria has integrated the fragment of the virus in its CRISP region represents a catalog of possible enemies, the DNA of possible enemies. The catalog is made of short fragments of different viruses, and actually this CRISP is translated. So from the translation of this CRISP region, you can produce small messenger RNA. Each of them contain 2 fragments. One fragment corresponds to the virus genome, Another fragment is part of the CRISP system, but not of the virus genome. This fragment, the small RNA fragment, sorry, I missed the problem, okay? This small RNA fragment is bound by the Cas9 protein. So there is introduced a complex between the Cas9 and this small RNA. Or there is another component of the RNA, that's just the details. So get this complex, and once the Cas9 RNA, CRISP RNA complex is made, this complex go through the cell scanning for DNA fragment matching exactly the fragment carried by RNA. So in principle, if here you have a sequence corresponding to the virus RNA, this complex is going around in the cell searching for virus DNA. And as soon as it found a virus DNA and it recognized the DNA because of the absolute complementarity between the DNA and the corresponding RNA present in the Cas system, the Cas enzyme simply cut the DNA. And because it's a virus, as soon as the DNA is cut, the virus is dead. This is a perfect immuno-like system for bacteria. About 10 years ago, two scientists, Jennifer Doudna and Emmanuel Carpentier, they had the idea to use the same system, replace the CRISP sequence from bacteria with a target sequence made by scientists, and use the the CRISP Cas9 system to tag specific sequence in the eukaryotic genome. And this is presented in the next part of the video. Okay. So there's the method has been slightly modified. So there's been some change to make it more easy and more simple, the production of this so-called guide RNA. Now we call it guide RNA, the RNA which correspond to the target region you want to deal with. And you can make a complex between the guide RNA and the Cas protein, and you can introduce this complex in the eukaryotic cell. Actually, this complex search for the corresponding genome DNA sequence, and if you design the guide against the sequence of a specific gene in your genome, the CRISP Cas9 complex will search for this corresponding sequence, and as soon as it reaches the corresponding sequence, the Cas9 simply cut the DNA. Sorry again. Simply cut the DNA because, well, in eukaryotic genome, as soon as you have a damaged DNA level, there are some repairing mechanisms that take care to repair the DNA. However, these repairing mechanisms are error prone. That means that there is a significant possibility that a mutation is induced during the reparation, and this is presented here. Okay, the repairing mechanism acts, replace the continuity of the DNA, but there are some mutation. This mutation allow to make can be selected by the scientist and used to introduce new characters. Okay, so CRISP is a natural mechanism. Here is a list few key features of CRISP. So CRISP is a system for cutting DNA in a position defined by a unique sequence. It's been evolved from bacteria to destroy viruses. In this sense, it's sort of a natural mechanism. In the commercial version, the cut point is predefined by the operator. It's not random. Following the cut, the natural repair mechanisms are activated, and the repair can produce small mutation, mainly short insertion or deletion. And this can alter the open reading frame of the gene, and you can have a stop codon, for instance, so you can change the sequence. And then there are many additional versions of the CRISPR-Cas system that allow to make also base mutation or changing specific base and so on. But in principle, you get a mutation. Of course, when you cut the DNA, you can use the system to insert an exogenous gene at the potential predetermined point of the genome. The system works in all eukaryotes. For instance, it's used also in medicine for to combat genetic diseases. That is very important. The system requires an excellent knowledge of the genome and of the gene function. This is a really important point, and this is why we are still pushing energy in genome sequencing, not only to sequence the reference genome for each cultivar, but also to have a representation of the diversity at the genome level. The approach is called pangenome analysis. Okay, so in principle, when you use genome editing, you can think to move from a standard random mutagenesis to a target mutagenesis. So when you do random mutagenesis, you take your variety, you add a mutagen agent, radiation, for instance, then you end up with 1,000 of plants, each of them carrying thousands of mutations in the genome, and you have to search for a mutation in your specific gene. If available, not necessarily the mutation is available. Not necessarily it's possible to work with thousands of plants. If you think about fruit trees, for instance. Well, mutagenesis in fruit trees is getting very problematic simply because you need to grow thousands of plants, search for thousands of mutations and screen them. But if you are lucky, you may also be able to find the mutation. Then you have to make select the plants carrying desired mutation, and you need to go for a cleanup of the genetic background. So you want only one mutation, not the mutation in another gene. And there are systems, for instance, you can do it by tilling or by simply looking at the phenotype. But certainly it's very difficult to have a plant with only a single mutation. Most of the time you get other mutation near around the selected mutation. This landscape is completely changed if you think about genome editing. Genome editing using CRISPR-Cas9 simply allow you to get a mutation in a specific position predetermined by the scientist. That is very important. You can get a mutation in any gene of your genome. What you need to know is the sequence of the genome and possibly the function and the exact function of the gene. So if you compare this, the genome editing with the traditional transgenesis, it's clearly very different. Genome editing is an editing. You correct a DNA sequence. Transgenesis, by definition, you take a gene, very often a chimeric genes made of two components, promoter and encoding sequence, for instance, coming from different organisms, and you put your transgene into another crop without any limit in terms of transferability. Between those two technologies, there is another technology, as I told you, in Europe, it seems to be very relevant. It is called cisgenesis. So cisgenesis is a transgenesis with some limitation. When you do cisgenesis, you take a gene, an entire gene, meaning including promoters, coding strands, and everything, and you take the gene from one variety, you introduce the gene into another variety of the same cross species. Or you can take a gene, for instance, from a wild plant of a species and you introduce to the cultivated variety of the same species. The principle is that the donor and the target organism must be fully interfertile. So it could be made also by cross. Why this is very relevant, at least in some situation? Because in principle, when you do back cross, it is the standard approach to introduce a gene from a wild plant into the modern plant, to a variety. What you get at the end is a variety which contains not only the gene you wish to be introduced, but also there is some linkage drug. So some DNA which is around the gene to be introduced is goes together with the gene which has been selected. In this way, the final variety is never 100% identical to the recurrent parent. It's very similar, but you can have 2, 3, 4, 5% of the genome from the donor parent. If you do a cisgenesis, only the gene of the resistant variety, only the gene of interest is introduced in your elite variety. And for instance, this is an approach that can be extremely useful and powerful to solve some situation, particularly if you work, for instance, with fruit trees. I will give you an example later on. Okay, so there is a problem. Nothing is perfect, probably, but if you make, as I told you before, the genome made the plant is a plant that has only a mutation in the genes. It means that it does not contain any transgene. But of course, when you do genome editing, first of all, you introduce in the DNA of your target plants a construct using standard transformation technology, and this construct carries the Cas9 gene, as well as the guide RNA. Then you need to find a way to eliminate the cassette with Cas9, RNA guide, and eventually selection marker. I have some background, somebody speaking. No? Okay, so there are some approach to segregate out this cassette. The first, in many crop, particularly in the annual crop where cross is can be easily made, you can segregate out the cassette with the Cas9 and marker simply during the next generation. So you get the plant carrying the mutation and the transgene, they can be segregated according to the Mendel law. And this is the most easy way. If you work in tomato, wheat, this is the standard one. But there are some problems. For instance, for a vegetatively propagated crop, think about an apple or a grapevine. In this crop, it's very difficult to get a segregation because if you make a next generation, what you have is not the same plant that you have at the beginning. So there are systems that allow to eliminate the cassette. One is the most famous one is the Cray-Lox recombination system. Cray-Lox is a specific recombinase technology that produces a deletion. And if the region deleted contain the Cas9 and marker cassette, then you are clean your plant. However, there are small signature DNA signatures are left in this process. Or somebody is now thinking to use protoplast and using to make genome editing protoplast using purified ribonucleoproteins, Cas9, and make a regeneration from protoplast. This should give a plant which is completely exogenous DNA-free plant. This again is very important because the public opinion really does not like, at least in Europe, to have a plant or a food carrying a small part of DNA coming from something else. This is a matter of discussion at European level at this moment. Anyway, technology are available. And last technology, for instance, is a great interest if you work on grapevine. Okay, so this is my introduction. And now I will tell you more about the experience we made in Italy during the last 5-6 years. The Italian government in 2018 has approved a national project for plant biotechnology. The project is called Biotech. not so original, the name, I apologize. But this project was at the funding of 6 million euro and was considered a strategic program for the implementation of plant breeding innovation in Italy. The project actually has lasted till the end of last year. But biotech was funded by Italian Ministry of Agriculture and was coordinated by CREA. CREA is the Agricultural Research Council of Italy. 6 million euro, most of them for CREA, but we also had some money for other research institutions, particularly university. And by definition, we say biotech used only genome editing and genesis in crop plants to develop new knowledge, gene function, and select new genotypes. It was the definition of the program. Biotech was organized in 13 research areas. Each of them were focusing on one or few crops. So in total, about 15, 16 different crops were studied using genome editing and genesis. The idea was to develop a know-how on the most relevant crop for Italy. And then once we have the know-how, try to develop some application. So you will see in the next slide how, depending on the crop, we use different strategies. Sometimes we use genome editing, sometimes we use genesis, and we work on different traits. Just keep in mind that the idea behind the project was to develop a know-how. So the aim was to develop a national know-how capable to operate on many crops, possibly most of the Italian relevant crops. And then, because at least in Italy, the public opinion 6 years ago was not generally in favor to biotechnology, we put a lot of effort in biotech to promote a favorable environment for plant breeding innovation in Italy, and promoting a science-based awareness of plant breeding in the society. This, I think, during the last 5 years, also due to the impact of this project, the feeling is changing in Italy. So now people are less aware of plant breeding, are less afraid of plant breeding. They start to think that plant breeding is not so bad as they believe 10 years ago. Okay, the other point is biotech was prepared based on the current legislation. And that means that although we develop many potentially interesting plants, so far we didn't make any field trials for genome edited or transgenic plants. Since a couple of weeks ago, it was not possible, let's say, theoretically it was possible, but from practical point of view, it was not possible to make any kind of field trials for GM plants or NGT plants. This because the regulation in principle was accepting field trials, but it was so complicated to fulfill the requirement that nobody in 20 years succeed to fulfill the requirement to have any kind of field trials for GM, for instance, in Italy. Now, last week, the national regulations changed a bit, so we expect to be able to do some field trials in the next year. But when the project was written and when the project was run, there was no option to make any kind of field trials. So what are the examples from biotech? I will first, in this introductory slide, to just simply to get the feeling of the approach we use. But we use genome editing and also we use genesis, but working at least 3 main levels. First approach was dedicated to editing genes coding for enzyme parts of a metabolic pathway. So the idea is to modify a metabolic pathway, and this modification may have led a change in the phenotype, sometimes simply an accumulation of specific metabolites. That can be, for instance, antioxidant metabolites or metabolites of interest, or simply if you stop, if the metabolites we are dealing with is a hormone, then get an impact because the hormone changes the situation in the plant. Another approach was based on editing negative regulators. There are many processes in plant life that are controlled positively by activators and negatively using negative regulators, repressor. If you knock out a repressor or a negative regulator, the process is upregulated. So there's a lot of examples in the biotech field dealing with knocking out negative regulators, and the knockout of regulators can have a positive effect. Probably the most famous example known since the 60s is the gene called MLO. It's a gene conferring resistance to powdery mildew in barley. A knockout of this gene makes the plants resistant. This is a typical example of a susceptibility genes. If you knock out the susceptibility genes, plants become resistant. It's a sort of negative regulator. The third approach we follow in biotech was to introduce new gene from wild relative or closely related species. This is an approach based on cisgenesis. The idea is to take a gene from a plant, introduce this gene into another variety of the same crop, and this sometimes, as I told you already, can solve some relevant problem. Next slide, I'll give you some example. First example, simply to give you the taste how different can be the problem that can be solved using new breeding technique or new genomic technique. First problem I try to give you is this one. So broom rapes, or banche in Latin, broom rapes are a parasitic plant of tomato. Broom rapes are parasitic plants. They do not have green leaves. They simply take the nutrients from the tomato plants. And the contact between the parasitic plants and the tomato plant is at the root level. This is a tomato plants infected with broom rape. And there is, if you have this problem, the yield is significantly less because the impact is very high. From biological point of view, there is a molecular dialogue between the broom rape seed and the roots of the plants. Broom rape seed germinate only when a root from tomato grows very close to the seed. And the seed can, feel the tomato roots growing, because tomato roots, when grow, they make an exudate. And this exudate contains a compound called strigolactones that are perceived by the seed of broom rape. When a broom rape seed touch a strigolactone, start to germinate, and then reach the root of the plants, make an haustoria, and then start growing, taking all nutrients from the tomato plants. Strigolactone is a kind of hormone which controls different activity in the plants, but also the germination of broom rape. To make a plant resistance to the germination of broom rape, or if you like, to make the broom rape blind, so in a way that the broom rape cannot see anymore the plants, you could take advantage of the knowledge about the metabolic pathway leading to the production of strigolactones. There are several key genes here indicated, and if you knock out using genome editing some of those genes, you may think to have much less production of strigolactone. And if there are no strigolactone released in the soil, most likely your broom rape cannot germinate, cannot see the tomato. Okay. A colleague of mine working in this biotech project simply make a mutation in 2 or 3 genes controlling strigolactone biosynthesis to limit broom rate germination. And this is the result they got. So this is the knockout for one of the gene called CCD7. This is CCD8. And this is the percentage of broom rape germination when the tomato plants are grown together with broom rape seeds. And you see that there is a significant reduction in the capacity of broom rape to germinate if the plant does not produce strigolactone. Perfect. This is absolutely a good result. I have to give credit to the CREA Horticultural Research Center, and particularly to Alessandro Nicolia and Teodoro Cardi, that did this job, and they produce indeed a tomato plant which is resistant, or at least is invisible to broom rape. These are the phenotype of the plant with different knockouts in different genes. Clearly, some of those plants has also a change in the phenology, in the phenotype, because the strigolactones are hormone, so it may change, but not all genes have the same impact on the plant growth. This one, CD27, is very similar to the normal plants. Again, there is also a trade-off between the growth capacity of the plants and the yield, but actual yield seems not to be affected so far, and the capacity of the plant to resist the broom rape. But this is a clear proof of concept that you can block germination of broom rape, making tomato invisible to broom rape. Well, this is another completely different story. Again, we're dealing with a metabolic pathway. Again, we use genome editing to stop a specific and to knock out a specific enzyme in the metabolic pathway. And this modification in the enzyme does not block the metabolic pathway. But what we are interested in now is not on the absence of the final product, like in the strigolactone story. Rather, we are interested in the accumulation of the precursor. This is a story which has been published in Natural Plant. It's a collaboration between the John Center, the CNR in Italy, and the CREA, my research center, Genomic Bioinformatics, and Fabio D'Orso is the scientist working with us in Rome. And in this work, they simply follow a pathway in tomato. The pathway moving from the hydrocholesterol down to alpha-tomatin, which is an alkaloid of tomato. This is the phytosterol pathway. And if you knock out a gene called 7-dehydrocholesterol reductase, what you have is okay, there is no accumulation of alpha-tomatin, but this is it's not relevant for us and for tomato as well. But you have an accumulation of the precursor, the 7-dehydrocholesterol. If you take a tomato plant with high level of 7-dehydrocholesterol and you expose the plants to UVB, the 7-dehydrocholesterol can be transformed in vitamin D3. Vitamin D3 is the precursor of vitamin D. So you can have a tomato fruit that is able to accumulate a precursor of vitamin D3, of vitamin D. And this is a very interesting application, because using simply a knockout in a single gene, we produce a completely different compound that is relevant at nutritional level. Simply keep in mind that as a standard, vitamin D is synthesized or is accumulated only by animals. So you usually take with D vitamin from milk, from meat. Now you can have D vitamin also from a vegetable. Okay, let's change again the perspective. Also in this situation, we work on a metabolic pathway, but we work on a metabolic pathway while actually is a negative regulator of a normal dedicated pathway. But the problem is completely different. So we now work on eggplant. Eggplant, there might be a problem because we wish to improve harvest and the storage capacity of the eggplant fruit. And you should keep in mind that eggplants, when they are harvested before physiological maturity, and seed in the eggplant fruit are still unripe. Then when you during commercialization, during the shelf life of the fruit, the maturation process proceeds, and when the fruit are completely mature, and the product is no longer on the market, so it's not really, cannot be eaten anymore. So when the eggplant inside is dark, you discard the eggplant because it's already gone. But this is the physiological maturation of eggplants. Might be a solution. You can make fruit without seed, so parthenocarpy fruit. This allows to extend the harvest and the marketing time of the eggplant, because the maturation process can be slow significantly. So you basically extend the shelf life of the product. How can be possible this? Well, it's easy, because eggplants fruit result from an ovary growth following pollination. After pollination, the block of the auxin synthesis is released, auxins start to increase, and fruits develop. But this block of the auxin synthesis is a typical example of a negative regulator. There are a number of genes that negatively regulate the synthesis of auxin in the eggplant's fruit. So if you knock out a regulatory gene like this gene here, IIA9, there is no repression of auxin biosynthesis in the after flowering, so in the ovary. And if there is no repression of auxin synthesis in the ovary, plants move directly from flowering to fruit development, and you get the fruit without seed. And that is possible here. You can get fruit, an eggplant fruit without seed. This should extend the shelf life of the fruit, and certainly should make easy the harvest, should extend the harvesting time of the plants. This is the work was carried out in my research center, Create Genomics and Bioinformatics, by Giuseppe Rotino and Laura Toppino. Again, we can change another topic again. We can, again, to give you the feeling that this technology is really powerful. It can be applied to many different situations. Now we are talking about basil. Basil is a traditional plant in Italy. It's the plant that is used to make pesto. And so there is a large industrial interest in basil. And basil has a problem, is that most of the basil variety are susceptible to downy mildew. So there is an interest to make a plant genetically resistant to downy mildew. A possibility is to knock out a gene responsible of the susceptibility to downy mildew. Exactly the same example as MLO in barley. This gene is called DMR6 and is being described in different crops, in different plants. This is the paper I show here is from Arabidopsis. And in any crop where DMR6 is knocked out, you may observe an increased resistance to downy mildew. And this is also the example I showed you before, here in this slide. So we had a number of plants carrying knockout of the MR6. And see the wild type plants as a, this is the percentage of infected area, so it's severity in leaves after artificial inoculation with downy mildew. You see the wild type has a 100% or almost 100% infection already after 10 days after infection. If you take this plant here, 22B, which is a knockout plant for the MR6, you can see that the infection, the spread of disease is much slower, and clearly knocking out these genes confer a significant level of tolerance or potential resistance. These are the figures of the different leaves. Again, you see the 22B here, the leaf is absolutely intact, almost no disease after 10 days of after inoculation. So again, the knocking out this gene is something makes sense. And this work was carried out by Marina Laura and Marco Savona in the CREA Research Center for Horticulture Crop. Another example. Okay, now I move to other two examples where we use cheese genesis. So the idea to take entire genes from one accession and transfer this gene to a completely different accession, the same crop, or in any case, through the donor plants and the receptor plants are fully interfered. This is an example of a gene present in bread wheat transferred to durum wheat. So bread wheat and durum wheat are two closely related species. So they are two different species. However, under some condition, durum and bread wheat can be crossed and genes can be transferred. And there are examples in the literature of genes transferred from durum to bread and vice versa. So it's really possible. It's not very easy, it takes time, takes a lot of back crosses, but it's technically possible. So this is when this is advantage. Why we need to cross, or why we need to use transgenesis or cheese genesis to take genes from one crop to the other one? Because there are genes that are present only have been evolved only in durum or in bread wheat, and those genes could be useful in both species. An example of this gene is a type of gene is a gene called LR67. That is actually a sugar transporter, and the mutation in this gene confer a durable, no race-specific resistance to a number of diseases, particularly Pauli mildew, leaf rust, yellow rust. It's not a complete resistance, it's a tolerance, but again is significant because it's no race-specific. It's not going to be overcome by a specific race, for instance. What we did during the biotech project, we simply take a gene from bread wheat and move the genes to durum wheat. LR67 is present only in bread wheat because it's carried by the D genome. And the D genome is the genome which is present in bread wheat, but is absent in durum wheat. And just remember that durum wheat is a tetraploid plant with A and B genome. Bread wheat is an hexaploid plant carrying A, B, and D genome. LR67 is on the D genome. So there is no option to find LR67 in the vast germoplants collection of tetraploid wheat. So there are, in principle, there are two strategies. As I told you before, you can make a cross between bread wheat carrying the A, B, and D, the D genome as the one with LR67. You can make a cross between bread and durum wheat. You can get an interspecific hybrid, which is has two entire genome and only one copy of the D genome. That is possible. Plants is viable. Then you can cross this interspecific hybrid with a durum wheat carrying a mutation PH1 gene. It's a gene which is involved in the recombination. I don't go for detail on this, but it's an essential component of the system. And then after many crosses, you can recover a durum wheat carrying a gene from the D genome. Okay, this is what you learn. This process takes at least 10 years, probably more. And it's really time consuming and there is a lot of work behind. Alternatively, you can take a genes from you can clone the genes from bread wheat, the entire genes, and transfer the gene directly through transformation in durum wheat. This is what has been done, and this is the phenotypic analysis of the plants obtained. These are this bar represents different level of infection and different scoring time for Pauli mildew. And there are some plants that has no infection, and some other very high infection. Then if you look for the plants carrying the mutation, see most of the plants with the cheese genesis, they have no, actually all plants with cheese genesis show no symptom. Again, the plants carrying LR67 gene from bread wheat are resistant or significantly resistant, in this case to Pauli mildew, or perhaps also to other diseases. This is a simple way to introduce a gene from a wild or from a closely related species to another species. And this work was carried out by Maria Massangio, Daniela Marrone, and Morelli, and that all of them work in the CREA Research Center for Cereal Crop. And now we go to the final example from biotech. We talk about grape wine. So grape wine is extremely important in Italy because it's probably the most cash crops of farmers that make a lot of money with grape wine. In Italy, wine is very important, and there is a lot of attention on grape wine. However, grape wine has a number of diseases, particularly downy mildew and powdery mildew are the major diseases for grape wine. And it's almost impossible to harvest a good grape without chemical treatments. And also in a biological grape wine, they are allowed to use some chemicals because again, it's almost impossible to get an harvest without fungicide. And because grape wine is one of the most relevant crop in Italy, on average, every year, about half of all fungicides sprayed in the Italian agriculture are dedicated to grape wine. So clearly, to improve the resistance of grape wine to disease has a huge potential impact on sustainability. Okay, new breeding technique, cisgenesis and genome editing, has the potential of eliminating most of the fungicide treatments in vineyards without change the quality of the product. This is essential because the value of the wine or the value of a vineyard depends on the quality. And now we have a system, the OC system, that make a certification, and they link the certification on the wine quality based on the specific cultivar that you use. So if you change a cultivar, you are not allowed to have the same certification. And then without certification, your income drops down. Okay, so it's very important to change the grape wine, well, to introduce resistance in grape wine without changing any additional gene. Okay, so what is strategy? Well, let's talk about downy mildew. Downy mildew is the most relevant disease in grape wine by far, and most of the fungicides are used to control downy mildew. However, there are in a wild grape wine accession plants showing resistance or carrying genes confirming resistance to downy mildew. Of course, this is a wild grape wine, so of course you will not be able to get any kind of wine from this kind of plant. Nice to see, but absolutely awful to eat and to use to make wine. But this plant is interfertile, can be crossed with a normal grape wine, the most nice grape wine cultivar used in Italy. And so in principle, the two plants are interfertile. And in the past, somebody has tried to use a backcross approach to select new grape vine varieties, resistant grape vine varieties. So the plants can be resistant, but because of what I told you at the beginning, those plants cannot be considered identical to the traditional plant. And because they are not identical, they cannot grant the same certification. And if you don't are able to grant the same certification, your income drop down. If you look in the internet, you find this brand, PU International, for instance, a company is marketing already grape wine resistance to downy mildew generated through backcross from standard variety with wild varieties of grape wine. This is possible, it's been done, but again, this grape wine cannot be granted the certification. The approach is using cisgenesis, making resistance the traditionally used grape wine variety. So now we are in the process to use this, and There is a great contribution in this field by the group of Riccardo Velasco and Luca Nerva at the CREA Research Center for Viticulture, and they were able already to make a transgenic in vitro plants, cisgenic in vitro plants grape wines, so plants carry the disease-resistant gene. Of course, because in this moment we were not since 1 week ago allowed to make field trials with grape wine, we cannot prove that these plants in field are effectively resistant. Okay, but they contain the genes, they express the gene correctly, so we trust that they should be resistant once we will be able to bring the plants in the field. Okay, this is to give you an overview of example we produce using this kind of technology to change the crop currently used in Italy. Probably most of those crops can be of interest also for Turkey. The biotech project indeed made also many other work, and some of them are still in progress, some are more advanced, some are less, but no, in more than 15 crops, we were certainly able to develop the know-how and technology for genome editing and cisgenesis. And this is a big results to my understanding. Okay, now I spend still 5-6 minutes I'm talking about the regulation. Europe is particularly has a very severe regulation for GMO, and there was, as was also mentioned before at the beginning, there is this directive called Directive 2001/18 that was approved by the European Parliament in the year 2001 that regulates the cultivation and the use of GMO plants. So the same regulation, the same law regulate both the cultivation in the field and the use of the GM products in the market. This directive has been designed to control GM plants. And there was a time when in Europe, most of the public, of the general public was strongly against GM plants. And most countries are still very against GM plants, particularly by Italy, but also the Nordic countries, for instance. So there is a strong movement, green movement, which is against biotechnology by definition. So the law was very restrictive. What is come out now, after 20 years, is that this law, also apply currently, at least presently, also apply to genome editing and cisgenesis. And why it applies to genome editing and cisgenesis? It's a matter of definition. I think we need to understand this step very. I mean, I spent some time to present you, because if you don't understand this question, then it's difficult to understand how it's difficult in Europe to change the law or to modify the system. So this law starts with a definition of what is a genetically modified organism. The law says GMO means an organism, with exception of human beings, which the genetic material has been altered in a way that does not occur naturally by mating or natural recombination. Okay, this definition is extremely wide. Almost everything belongs to this definition. Many different methods, breeding methods, can be fit in this definition. It's so wide that the same law, the same directive, at the article number 3, defines some exception. Okay, so there is an article in the law saying that this directive shall not apply to organisms obtained through techniques of genetic modification listed in Annex 1A, 1B. Let's go to Annex 1A, 1B. 1A lists the modification that are not, or technologies that are not considered genetic modification. And what is indicated mainly is polyploidization. So if you do a polyploidization using colchicine, you double the genomic, the genome of a plant, for instance, that is not defined genetic modification. Okay, so that is an exception. So the law does not apply to this kind of situation. You do polyploidization first. In Annex B are listed technology that are genetically modification by law, but they are not. They are exempt to the application of this directive because long-lasting. So because there is a long experience, that is the definition, the official definition. So the law says that mutagenesis or cell fusion, for instance, plus fusion. So mutagenesis is a genetically modification. So by law, a plant obtained by mutagenesis can be considered a GMO. However, because mutagenesis is largely used since 50 or 60 years, can be considered safe. And because it's considered safe, this directive does not apply to plants generated by mutagenesis. Okay, nice. But what we have now is a genome editing make in principle a mutagenesis. As I showed you before, the edited plant at the end of the process is a plant carrying a mutation. And if you remove the cassette with the Cas9 and the selective marker, at the end of the process, the plant's carrying mutation cannot be recognized cannot be found different from a natural mutation. So natural mutation, GM, and genome editing mutation are exactly the same. However, when this question was brought to the Court of Justice, the European Court of Justice, a couple of 4 or 5 years ago, the Court of Justice simply said that genome editing, being a mutagenesis, can be considered under the umbrella of the law. However, because it's not listed in the Annex 1B, cannot be exempt from the application. Because Annex 1B mutagenesis is a technology that produces a GM plant, but because it's been used since many years, is safe and therefore is excluded by the application of the law. Genome editing is a technology to produce a mutant, therefore is a GM by law, and because it's not listed in Annex 1B, goes within the law, cannot be used without the law. So this is now there is a strong discussion in public opinion in Europe. The general feeling is, I would say, I can be optimistic, but nobody knows exactly how it will end. Starting from next month, the issue is in the European Parliament, so will be discussed in the European Parliament, and we expect to have a new definition, a new law, or a new regulation for genome edited plants, hopefully before the end of this year. But I'm not capable to make prediction on the political issue, so I have to wait and see what happens. But this is the current situation in Europe. And if you compare Europe versus the rest of the world, this is the situation. So the country indicated in green are countries where GM plants, where genome edited plants are considered normal plants, assuming that there is no exogenous DNA. So if you can prove there is no exogenous DNA in most of these countries, plants can be considered as a normal plants. Europe and also New Zealand actually are the 2 parts of the world where still there is no law in this direction. As I told you, all scientists in Europe really hope to have this technology ready this technology ready to be used by the end of this year. Actually, I also see that Turkey is not listed in this. I apologize, I don't know about the situation in Turkey. I have a last slide, and this is another topic, still regulation, and this is something which, at least from a European point of view, is very relevant, or will be relevant. Because when you do an edited gene, most of the time the gene can be patented. So you have a patented gene in a genome. How this can conflict with the breeding rights? So breeding rights, for those of you who doesn't know what is the right of every breeder to use any registered variety as a parent for a new breeding program. So every breeder can take a registered variety from the competitor, for instance, and use this registered variety in the breeding program. If you have a variety which contains a patented genes, there is a patent on a gene, but then the genome is not patented because the genome goes through with the breeding rights. So a company has to be free to use the genome and may talk with the company who patented the gene, the mutated genes, to use the gene. In Europe, there is also a lot of passion, say a lot of concern about the possibility for small breeding company to assess this know-how, to assess the patented genes. So what I would like to tell you here now, and that recently there has been an initiative in Europe that can try to make an answer to these questions before then the question become too loud. And this initiative is called, it's made to conciliate the access to the patented genes and the breeding rights. And if you go to a website, you can find this initiative called Agricultural Crop Licensing Platform. And this is an initiative to facilitate the access of patented traits. It's still not active, but has been, to my understanding, is the best proposal I found to solve this. Well, might be a problem in the future. I'm sure it will be a problem in the future. So the idea is to provide to a company that is interested to use patented genes, or also plants carrying patented genes in its own breeding program, could register on this platform and then simply notify to the other company present in the platform that they are using a specific variety. And if they still will keep the patented genes, then they have to pay something to the company who has patented the genes. But if they use only the genome without the patented genes, there should be no problem. And this is the website, so you can go and search to this website for your convenience. And this is the list of the company who developed this proposal. You see, there are many large international companies like Corteva, Bayer, Syngenta, but there are also breeding companies like Limagren and KUS, and also small breeding company, the one here on top. And this is mainly from Germany, Denmark, I think, and some few others. So this is a proposal, still not active, but I would like to bring you this issue because I guess that the conciliation between breeding rights and the access to the patented gene is something that will be very relevant for the public acceptance of this technology, and also for the capacity of small, medium breeding enterprise to work with this innovation and to not to be put apart. So not to exclude those companies in a niche market, but simply to take advantage from the research carried out by other companies and make its own breeding variety, including also patented genes. And that is all from my side, and thank you for the attention. If there are any questions, I would be happy to solve. And I apologize to the interpreter if I've been too fast. I try to remember every time to be not too fast. I don't know how it was.
It was a beautiful presentation. Thank you, Professor Dr. Luigi Cattivalli, for sharing your insights on the National Biotech Project. Now I would like to move on to the Q&A session, where I will present the questions to you. The first question is coming from Professor Dr. Gültekin Yıldız. Can CRISPR-Cas9 cause interventions outside of the target region? And if so, how can they be determined technically?
Okay, the question about what we call off targets. So the possibility that the CRISPR-Cas9 complex, instead to target the gene of you wish to, goes to another sequence. Well, there is some possibility. This is not absolutely safe, but there are two components you should consider. The first one, again, you need a very deep genomic sequence of your crop. because if you know exactly the genome, if you know exactly the genes, it's much easier to design a specific guide RNA, where the specificity is really the key issue. I also suggest that before I go for the market or to the field trials, any kind of plants can be sequenced. So now sequencing a plant is really relatively cheap option, and if you sequence the plant, if you have an off-target mutation, this off-target mutation could be easily identified through a sequencing approach. So there might be a problem, but there are plenty of solutions for this problem. I mean, we should not be aware. I mean, plants is not a human being. If you don't like your plant, simply you kill it, you cut it, and then you make a new one.
Thank you for your answer. The second question is coming from Professor Dr. Kenan Turgut. Can anybody use CRISPR-Cas9 system commercially?
Sorry, anyone use CRISPR-Cas9
system commercially?
Yes. I mean, what I mean, there are also websites where you can buy the constructs. It's very easy. I mean, you buy a construct and then you can do it in your lab. It's not so difficult to make a transformation, and certainly you can use commercially. Yeah, but there are also companies who simply provide a service. If you're not willing to do yourself, but you know exactly what kind of genes you want to modify, you can ask a service company to make the work for you. Again, what is fundamental, there are two components that are really essential: the knowledge about the genome and the gene, and then the transformation technology. Because one of the limits you still have in some plants, think about some fruit trees plants, is olive trees, for instance, is the transformation. The limit of apply genome editing in olive trees is not genome editing technology per se, is a transformation protocol for olive tree, which is not yet available. And I guess in Turkey, olive tree is an important crop.
Thank you. The third question is from Dr. Hatice Çölgeçen. Dear Dr. Kativalli, thank you for your presentation. How do you think the future for agricultural products in functional food in innovative technologies in plant breeding?
Well, how do I see the future is I don't know if I can be able to predict the future. What I think, I mean, I can tell you the experience in Italy. Okay, this is something which is history. So what really make the Italian Parliament more open-minded about genome editing has been the last 2 years, where we had a strong impact of drought. So drought in last year, 2022, and this year, but now it's a completely different situation, but certainly in 2022 was very severe in Italy. And this year, the beginning of the year was very bad. Under this situation, after 2 years like this, the public opinion and the member of the parliament realized that we need to do something. Therefore, they now make a specific law to allow field trials for genome edited plants that can contribute either to drought tolerance or to disease resistance, because the agricultural sustainability is a big issue in Italy, and reduce the chemical input in agriculture seems to be mandatory for the public opinion, and therefore you might need a solution. If you cannot use pesticide, you need to use genetics. So plants can be resistant because you protect the pesticide, or because you make the plant genetically resistant. And the example of the grape wine I showed you before, this is a great example, because in Italy, half of the fungicide goes for grape wine, and make grape wine resistance to disease is a lot of could make a lot of impact in terms of sustainability. So I see the future. Well, there'll be a solution to have plants with higher antioxidant content, more nutrients, and so on. Yes, but I think at least in Italy, the genome editing will penetrate in the plant breeding in the society, mainly because of the issue against chemical input in agriculture or drought tolerance.
Thank you. The fourth question is from the Dr. Fatih Sezer. Thanks for the presentation. Regarding parasite-resistant plants, do you think targeting strigolactone transport to modify exudation could also be a viable approach?
Sorry, I mean, I missed the first part of your question, sorry.
Regarding the parasite-resistant plant, do you think targeting strigolactone transport to modify exudation could also be a viable approach?
Yeah, yes, there are some more, I mean, after we have proved the principles, I mean, we have proof of concept that modified strigolactone is a solution, a possible solution. Yes, we can now be more precise in the system. There are genes controlling the transport of strigolactone out of the roots. That can also, I think that people working in this lab from CREA are also thinking to modify this to target this gene. Certainly can be a solution. So we need to fine tune the system, but clearly the pathway of the strigolactone is the one which is relevant and the one that has to be targeted. But yes, there are many options to improve the results. So then, I mean, another option, I mean, sometimes in tomato, they use tomatoes nested tomato. So you can use, if the plants, you can use, so actually if you make editing only the root and not the upper part of the plants, the upper part of the plants grow normally and the roots are resistant. But there are several options, but certainly we need to fine tune the system. That's your right.
Thank you. The next question is coming from Meryem Melisakar. Genetically modified organisms can be detected using molecular techniques. Is there a specific technique available for detecting gene editing plants?
Well, gene editing is a mutation. So if you do correctly the editing, you introduce a single mutation in a gene of target, then you remove the Cas9 cassette, a selective marker cassette, by segregation. For instance, if you work on tomato and wheat, it's very easy. You segregate, it's enough one generation. Then the edited plants simply carry a mutation. And because it's a mutation, is absolutely equivalent to any natural mutation. And if nobody tells you, if the company does not certify you that this plant has been produced using genome editing, nobody can understand the origin of the mutation a posteriori. Once it's been done, it's been completed the cycle, nobody can understand or can talk about the origin of this mutation. You will never be able to distinguish between a mutation generated by editing or generated by natural. I mean, I can tell you many examples. There are already examples almost on the market, but I'll tell you a very famous example from history. I mean, again, to the MLO genes in tomato, in barley. So MLO gene is a gene conferring resistance to polymedium. This gene was discovered in the 60s through a mutagenesis program using chemical mutagenesis. Okay, you get a gene mutation produced by chemical mutagenesis. 50 years later, somebody screening a germplasm collection of barley accession from Ethiopia find out a barley mutant carrying a mutation in the MLO gene and showing resistance to polymedium, exactly as the plants generated by mutagenesis. Now, if you make a genome edited plant, you can get a new mutation in MLO genes. You can produce again a plant carrying a mutation in MLO gene, conferring resistance to polymedium. So those three plants carry the same mutation, but from three different origins. Now, when you compare them, you don't see any kind of differences. So you cannot detect the origins once the process has been completed.
Thank you. The next question is coming from researcher Özgür Özmen. Sir, what do you mean full drum or bread wheat? Is it mean pure line?
What do you mean as
full drum or bread wheat? Is it mean pure line? They say.
I don't understand. What is this? Sorry. Durum wheat.
Yes.
Durum wheat.
Hello, durum wheat is the wheat you use to make couscous. Okay, you have couscous, you have burgul in Turkey. Burgul is made of durum wheat, and bread is made of common wheat or bread wheat. So two different crops, very closely related. So you can, there are two species, but you can cross.
Thank you. The next question is coming from Professor Dr. Bülent Uzun. Are cisgenic crops commercially marketable in Italy or in Europe, or are they treated like transgenes in Europe legislation?
At the moment, they are considered as transgenes, no doubt. I mean, we need to wait for the new law that should come from the European Parliament, hopefully before the end of this year. But in this moment, both genome-edited plants and cisgenic plants are considered as the same. Well, they are not separated. I would say they are not separated from GM plants from legislative point of view.
Thank you. This one is quite long. It's coming from Ayşenur Arslan. Dear Dr. Kativalli, first of all, thank you for your informative presentation. Do you have any studies associated with the increasing the specific protein expression in plants with plant biotechnological approaches? Or are you planning this kind of projects? As you know, plants contain multifunctional proteins. For example, hordein, that is a glycoprotein, evolves in barley, and these glycoproteins can act as a probiotic agent. Therefore, increasing protein expression in multifunctional proteins is a significant approach for agriculture and enhancement of using plants to produce functional food products.
I do not have an answer at the moment. Yeah, I don't have in mind an example. I would say, generally speaking, yes, why not? But simply, if you want to accumulate something, you can knock out a repressor, and then you get an accumulation of the product. So again, you need to know very well the function of the genes, the pathway you want to modify. And then for many, there are many examples, or there are many pathways which are controlled either by promoter, by activators, or by repressor. And there are many kinds of repressors, not only genes. You can use a microRNA to repress the expression of a gene. Again, there are many options. Certainly it's possible. I don't have an example just in front of me now.
Thank you. I will ask one more question. It's coming from Professor Dr. Sami Doğanlar. Did they test Orobench edited tomatoes with other soil-borne pathogens? Thanks for presentation.
Not yet.
Okay, thank you for your answer. And thank you for giving your time in this question and answer section. We greatly appreciate your answers. Thank you so much.
Thank you for this opportunity, and really have a nice work.
Thank you so much. And I would like to call our final speaker. Our final speaker is Professor Dr. Michael G. Thompson, the headmaster, Bashel Rice Chair at Texas A&M University and AgriLife Research in Department of Soil and Crop Sciences. He will be presenting on CRISPR-Cas9 editing technologies for crop improvement. Please feel free to ask questions in the chat section during the presentation, and selected questions will be addressed during the Q&A session. Over to you, Professor. Welcome.
Okay, thank you very much. So thank you also to the organizers for inviting me. I'm very excited about talking to this group. And also, it's excellent that I'm following Professor Kativalli, because he gave an excellent introduction, so it makes my job much easier to follow him. So I'm coming from Texas A&M University, Department of Soil and Crop Sciences, and I'll be also talking about CRISPR-Cas gene editing technologies for crop improvement. So for the outline today, I will just very briefly touch on an introduction to gene editing and some examples for CRISPR editing strategies for plant breeding and crop improvement. And then give an overview of the regulatory framework and consumer acceptance issues and IP landscape in the United States. And some examples of developing a crop gene editing pipeline at Texas A&M University in our crop genome editing lab. So thankfully, I don't have to provide much detail about the introduction. So we've already heard that CRISPR is a breakthrough gene editing technology. And we know the important aspect is that the Cas9 protein can directly target a specific DNA sequence for making a double strand break in that sequence. And so, of course, in 2012, that was the landmark paper from Jennifer Dunya and Emmanuel Charpentier, and they won the Nobel Prize for Chemistry for this technology. And so it's a very powerful technology. So I think we all know also that it's CRISPR gene editing is transforming science. And so we see many publications on CRISPR early on, really exponential growth in the number of publications, number of patents, and this just continues to this day. And of course, CRISPR is impacting a wide range of fields. And so today we'll talk about crop improvement applications. So looking at agriculture, I think there's a lot of interest in gene editing as the next food revolution, essentially making food healthier. So some examples, for instance, high oleic soybean oil, low gluten wheat, high fiber wheat, reduced allergenicity in peanuts, low glycemic index rice. These are just some examples of some of the healthier foods that are made possible through this technology. And so I think the next generation of gene editing products will be focused a lot on consumer traits, since GMOs previously, a lot of those traits that only help, producers really didn't capture, I think, interest in the general public. And so this new set of traits really are consumer focused, but also really can be used to help in global agricultural development. So really addressing global food security and improved crop resilience in the developing countries. So I'm just highlighting this article by Bill Gates on how CRISPR can transform global development. Gene editing for crop improvement could also help food security as well. So thankfully, I don't need to explain what is CRISPR. So if we all know that it's a bacterial immune system from invading viruses. So we can move on from there. The important part, of course, is that it makes a double strand break at a very specific location. So this is really above the PAM motif. So that for Cas9, it's a NGG. So the Cas9 protein searches for that PAM motif, and then upstream, it checks if the 20 base pair recognition sequence matches the guide RNA. And if it makes a perfect match or a very close match, it will cleave or cut the DNA and make that double strand break. So that's the important function of the Cas9, is making the double strand break. And what happens after that is the plants will repair that break. So when you have the DNA has double strand break, the plants have a repair machinery, and they will either use non-homologous end joining, NHEJ, or homology-directed recombination, HR or HDR, to repair that break. And so for us, it's, there's several applications. For instance, NHEJ, normally it's a simple, you can think of it as a fusion of the DNA, but it's also error prone. And so often there'll be a small insertion deletion that will occur that will essentially cause a mutation in the gene. And if it's in the coding region, it can actually knock out the gene function. So that's your simple gene knockout through small insertion deletions. Normally because of, it can introduce a frame shift mutation that can cause a premature stop codon that will essentially truncate your protein, okay? But at the same time, there's also gene insertion applications where if you provide a donor template, you can actually have either gene insertion or very precise gene modification. But these involve donor templates that have to be provided along with the Cas9 and the guide RNA. So those are quite a bit different, but also very useful for some applications. Okay, so just a little bit about how you actually do this in practice. So in plants, of course, as was mentioned earlier, you do need to know the gene sequence of your target. You have to design a guide RNA that has that 20 base pair target sequence that's immediately upstream of the PAM motif. And then you have to express that guide RNA in the plants. And so normally we'd have a small RNA promoter, such as U6, And then you'll have your guide RNA sequence and the single guide RNA backbone. And this backbone then with your guide RNA sequence can be expressed in plants. And at the same time, we have to express the Cas9 protein in plants as well. So this generally you'll have a single construct. So this is your plasmid DNA vector that contains a Cas9 protein and your guide RNA sequence. And then you can express it in plants. And so, of course, there's many ways to express, a transgenic construct in plants. The most common is agrobacterium transformation. And so this is really the most common way is using agrobacterium to transform. Essentially, it inserts the tDNA sequence into the plant genome. But there's also protoplast transformation and then callus bombardment and others as well. And so once the plant's transformed, it expresses the Cas9 enzyme, it expresses the guide RNA, and then these form a complex and actually will target the target sequence to make that double strand break. And so after that, you need to screen for the mutants. There's several ways. The most straightforward is just sequencing the target site so that you can visualize the mutation, but there's other shortcuts, for instance, restriction enzyme site loss, or the surveyor assay that provide high throughput methods to screen for your target mutation. So these are all being used for plant gene editing. So as was also mentioned, if you're starting a new gene editing project, you can actually order the plasmids online. So this is one of the most common plasmid repositories. It's called AdGene. And this particular one, you can go online and search CRISPR plasmids for plants. And actually, they have many different functions. So it's not just cutting. There's also like base editing and now prime editing and many different applications. So there's literally thousands of plasmids available that you can search from. And so when people usually have a publication, they'll just cite the AdGene number so that you can go to this service to request the plasmid vectors. And these will have the plant optimized Cas9 and various selectable markers and other tools available. Okay, so the other step is designing your guide RNA, and there's many online guide RNA design tools. I'm just highlighting one called CRISPR Direct. Essentially, you just insert your target sequence where you want to design the guide RNA. And you tell it, which plant genome you're using. So in this case, Oryza sativa, so the rice genome. And the reason to essentially give the genome is that you need to check for off-target effects. So to prevent really cutting in the off-target site, what these design tools will do, they'll actually search the entire rice genome and identify if there's any other locations that will have an exact match. And so in this case, there's only one exact match of the 20-mer, that's the 20 base pair recognition sequence. So this is a single copy recognition sequence across the rice genome. So we can use any of these for our experiment because this single copy is our target site. At times, you'll notice if there's multiple copies, you may have a greater chance of having an off-target effect. So it's important to use the online tools to design your guide RNAs so they're very specific. And also, they'll show here that you have the PAM site and your 20 bases. Often we'd like to have, roughly 50% to 60% GC content and other parameters to help ensure that the guide RNA will be working properly. But at times also, sometimes guide RNAs fail for unknown reasons. And so usually best practice is to design at least 2 guide RNAs for any target to ensure that you can knock out that gene target. So just briefly also mention that there is a non-transgenic approach to plant transformation or to gene editing in particular. And this is the ribonucleoprotein delivery, RNP delivery for DNA-free genome editing. And this was also mentioned earlier, but essentially you can even buy purified Cas9 protein commercially. You can also order synthetic guide RNAs commercially if you'd like to just essentially order these components and mix them in vitro in the lab and then directly deliver them to plant cells. So you can use, for instance, the gene gun, so particle bombardment to essentially directly deliver this complex, which is a protein guide RNA complex, into the plant cells. And once it's in the plant cells, it can then target the DNA and actually make the mutation. And then it's naturally degraded over time, because this is not inserting, there's no stable transformation involved. So there's no DNA that's inserted. It's only this protein guide RNA complex. So essentially, this is a non-GMO approach. I would say this is really less, not needed. I would say less useful in the US, since our regulations really allow for agrobacterium, based transformation. We just need to get rid of the tDNA insertion to make it non-transgenic. Okay. So that's the brief introduction. So I will next go to examples of CRISPR editing strategies just from, the field. And, as was also referred to earlier, we have genetic tools for modern plant breeding. We have this genetic diversity, crop genetic resources. There's a lot of information on gene discovery and allele mining. And so this helps us, for instance, we can do marker-assisted selection and genomic selection. Of course, these are non-GMO. We also can do genetic engineering. So this would be transgenic approaches, which of course would be a GMO product. And then in between, we have genome editing. And so in many cases, genome editing can be non-GMO as well, as was talked about. So it's essentially a mutagenesis approach that's targeted and very precise. But I would mention that that's not true for all genome editing. There are applications, for instance, inserting genes where it can be a GMO, right? And so this is using, again, kind of the terminology from the United States, where genome editing can be non-GMO or GMO, depending on whether it has transgenic insertion in the plant genome. Okay. So just a few examples from the literature. Early on, many people were targeting phytoene desaturase, PDS gene, because it gives a nice albino phenotype. So this is rice. If you knock out PDS in rice, you have an albino seedling here that's kind of stunted. So it's a nice visual phenotype that you have successfully knocked out the PDS gene. And of course, if you sequence it, you'll notice that most of them are small insertion deletions. So again, if you have a one base pair insertion or one or two base pair deletion, you'll have that frame shift, which knocks out your protein function. Now, other studies have really gone farther than that and actually have looked at multiplex gene editing. So in particular, there's this publication from 2017 where they targeted actually 8 gene targets in rice and actually designed guide RNAs to knock out all 8 gene targets. But I'll note that even though they targeted all 8 loci, generally they'll get a combination, they'll get different combinations of knockouts in any particular progeny. You'll see that potentially you'll have 1 knockout or 2 knockouts, 3 knockouts, and so on. It's actually quite difficult to knock out all 8 genes at the same time. You need a very large population. But in any case, many of these genes are well known in rice to have effects on grain size, on plant architecture, on heading date. So all of these can be then essentially just accelerating the development of variation by inducing mutations in these target genes. Okay, so another example, in this case using RNPs, so this is the non-DNA approach, is editing the ALS gene for herbicide tolerance. And this was a publication back in 2016, where just by essentially introducing several base substitutions, so it allowed this amino acid switch, which then actually converted the native ALS gene to allow for this herbicide tolerance in maize. And this is a common approach now, essentially making a non-GMO approach to herbicide tolerance, which many companies are interested in. And you may see, in reports that there are many other gene-edited products under development. These are just a few examples you can find in the literature. Early on, one study was looking at anti-browning mushrooms at Penn State University, and also many people working on citrus greening disease, for instance, in Florida, in the orange industry, to get disease-resistant citrus. And also fungus-resistant bananas and reduced gluten wheat. All of these are applications that are possible with gene editing. And you can find in the literature many other examples. So they range from disease resistance to different agronomic characteristics. In rice, we'll talk about grain quality examples and fruit size and flowering. So almost every major crop now has examples of gene editing for plant breeding, and most of these using CRISPR-Cas9. There's a few that have used the TALEN approach, which was an earlier technology, but CRISPR-Cas9 is much easier to use, and so it's more popular. Okay, so now I'll briefly just kind of talk about the regulatory framework. So in 2018, actually, there is USDA had their statement on plant breeding innovation, where they essentially kind of introduced this new principle that USDA does not currently regulate or have any plans to regulate plants that could otherwise have been developed through traditional breeding techniques. Okay, so as long as they're not introducing a new plant pest, essentially, if you have these particular what they call exemptions, then that could have been developed through traditional breeding, then they do not fall under any guidelines for GMO regulation. So for instance, deletions, so this would be a single deletion of any size in your genome, a single base pair substitution, or an insertion from a compatible plant relative, so in the same gene pool. And this is the cisgenesis approach that we talked about. And then the last one, they actually changed in the final ruling. So it's essentially these three exemptions. So in 2019, they had public comments. In 2020, they essentially finalized the rule, and which was called the Secure Act or Secure Rule of Sustainable, Ecological, Consistent, Uniform, Responsible, and Efficient. And if you go online now, this is, they say formally called Secure Rule. It's just the updated biotechnology regulations. And importantly, these are, again, three exemptions for plants modified in a manner that could otherwise be achieved through conventional breeding. So again, this is referring to the fact that any of these deletions could have happened naturally. And so there's no need to have further regulation if it's the identical mutation to a natural mutation. So these are the final ruling refers to a change resulting from the cellular repair of a targeted DNA break in the absence of an externally provided repair template. So this is again that, so say you have a deletion or a small random insertion that does not come from a repair template. So there's no, like we talked about before, there's no donor template involved. So the second is a targeted single base pair substitution. And then the third is the cisgenic approach. So introduction of a gene known to occur in the plant's gene pool, or a change in the targeted sequence corresponding to a known allele of such gene, or known structural variation, essentially. So that's, so for instance, if you take a gene from one rice plant and insert it into rice again, it's essentially from the same gene pool. That could have happened using conventional breeding, simply by intercrossing them. And so it does not need to have further regulations. Okay, so that's essentially the framework. I'll just mention that's the framework we're working on now. And that what it means is when you fall under one of these exemptions, that means you do not have to go through the deregulation process that you would with a GMO plant in the United States. So this allows for instance, field testing of gene-edited products. Essentially, we'll treat them just like any other, plant that could have been achieved through conventional breeding. So I will mention this, these don't cover all gene-edited products. It's still fairly restricted in that it's a single modification, for instance, a single deletion or a single base pair substitution. There's been some kind of feedback from the scientific community in the United States that this should be modified to allow multiplex editing, so multiple deletions or multiple substitutions, and that has, that has not happened yet. So we're still acting essentially under the single mutation rule here. But I will mention that if you make the single mutation with gene editing in one line, and then you make a second line that also has a single mutation, you can simply intercross the two to combine the two mutations. So you can ultimately have multiple mutations, but you just have to do the gene editing one at a time and then later combine them using conventional crossing. And that was actually from USDA-FAS. There's several other regulatory agencies. One of them is the FDA, US Food and Drug Administration, which also regulates plant biotechnology products, plants and animals that are food products. And overall, I mean, FDA regulates every food, whether it's a biotechnology product or not. They ensure the safety of food no matter how it is developed, essentially. But they have been working on updating essentially their guidelines to specifically address genome-edited food crops. But at the moment, there's really, I believe, no difference. Any food product needs to be ensured to be safe, whether it's biotech product or not. But I will mention that so USDA and FDA and also EPA on pesticides, they are coordinating. Essentially, they have a coordinated biotech framework that they're working on to continue to improve essentially the regulatory framework to make it easy to use new biotechnology technologies, but at the same time to continue to make sure that they're safe for human consumption or for growing in the environment. So that was on the regulatory aspect. Next is intellectual property. Of course, there's many patents that control kind of commercial use of the technology. So here's the rapid explosion since 2012 of patents. Of course, with patents, there's also many licensing agreements. So many of these kind of foundational patents are owned by various companies or, for instance, MIT Broad Institute. And they also had a dispute with UC Berkeley that's been mostly resolved. But essentially now all the companies then can license gene editing technology and then make it available, for instance, for human therapeutics or for lab tools or for agriculture. Specifically in agriculture, a few years back, DuPont Pioneer and the Broad Institute, so this is now Corteva, DuPont Pioneer is now Corteva AgroScience, kind of combine their IP portfolios to have a single kind of cross-licensing platform for CRISPR patents specifically for agricultural applications. So this brought together many patents for gene editing for use in agriculture. So if anyone wants to commercialize a CRISPR-Cas9 product for agriculture, they can go to this essentially kind of umbrella kind of organization then to cross-license so that you're free then to sell your product that resulted from gene editing. Okay. So I'll just kind of conclude with some examples from Texas A&M University on what we are working on in gene editing. And so, so one of the, well, first I'll just introduce my lab. This is the crop genome editing lab. We're really developing and optimizing high throughput CRISPR gene editing in plants. So myself and Dr. Septeningsi, about 5 years ago, we set up this lab, essentially piloting studies for gene editing across different crops, initially focusing on rice, but also we've done industrial hemp, cotton, sorghum, cowpea, and peanut. And then also researching novel methods for CRISPR delivery and implanted transformation. So I'll talk about those. But first, most importantly, you need a good plant transformation facility. And so we have what we call the multi-crop transformation facility at Texas A&M, led by Marco and Myra Molina. And essentially, plant transformation is very difficult for many crops. And so they've optimized agrobacterium-mediated transformation and regeneration. So for instance, cotton, potato, sorghum, rice. So this is the whole process of in vitro tissue culture and transformation and regeneration. And many times you have to essentially optimize each crop species independently, and also you have to improve the performance for each variety as well. So for instance, we have a Texas rice variety we call Presidio. And so that needed to be, again, optimized for the tissue culture and regeneration. So that publication was out, essentially making it more efficient to do gene editing. Now, I'll just briefly mention 4 examples in our lab of some of the research projects, mostly again in rice. So increased resistant starch, increased lysine levels, modifying flowering time, and exploring novel delivery approaches. So the first is knocking out starch branching enzymes in rice. So anyone familiar with rice, essentially the starch has different levels of branching, and that will determine whether it's amylose or amylopectin. So the highly branched starch grains that form amylopectin have quite a different texture and consistency than amylose. And so a very low amylose rice will be very sticky. So for instance, for sushi, what we call kind of waxy rices are very sticky, and high amylose will be a very kind of dry or fluffy texture. And interestingly, that one aspect of this is also related to what's called resistant starch. And so this is starch that cannot be digested in the human digestive system. And it's actually can improve the healthiness of rice and helping to actually decrease the glycemic index as well. And so there's been some interest in increasing the level of resistant starch. And for the most part, resistant starch is highly correlated with the level of amylose as well. And so if you look at trying to modify and develop high amylose rice that has higher resistant starch, you can actually knock out the starch branching enzymes. And so then essentially the starch will not form amylopectin, it will actually form amylose instead. Okay. So to do this, we targeted 4 starch branching enzymes in rice. So here's each of those 4 loci. And to first, essentially the first step was designing guide RNAs. And we have an in vitro RNP assay, essentially, to confirm if the guide RNAs are actually working. So essentially what this does is you can, in vitro, you have a PCR amplicon, and you introduce your Cas9 guide RNA complex. And if it cuts that PCR amplicon correctly, you'll see 2 products here. Actually 3, you'll have your 2 cut products and then the original that still remains. And so this shows that you have successful cleavage of the DNA product. Okay, so this is an early indication that your guide RNAs are designed properly. And so this work was performed by Sudha Biswas in our lab with Dr. Septaningsi as well. And essentially what they found is several of these knockout lines had 10 to 15% higher resistant starch, or actually 10 to 15% resistant starch content, which is much higher than the wild type, which has very little resistant starch, and up to 30 to 42% amylose content, which again is much higher than the wild type, which was under 20%. So these are both high amylose and high resistant starch lines. I will mention that some of the lines had relatively high amylose, but low resistant starch. So there is some complex relationship between the percent of resistant starch and the percent of amylose. But it is very promising that several simple gene knockouts could actually impact something as important as your amylose content. Okay, so the second example I'll talk about, and this was actually referred to previously as a way, if you knock out one of the enzymes in the biosynthetic pathway, you can actually increase the production of one of the upstream products because it's not being essentially transformed into something else downstream. So what we're looking at here is this is the lysine content in rice grains. It actually goes through this biosynthetic pathway, and there's two enzymes downstream of lysine, LKR-SDH, that if you target those, you can actually have it accumulate higher levels of lysine in the rice grains. And the wild type has relatively low levels of lysine to start with, less than 4% in most cases. So this experiment was done by Khushbu Rastogi, a student, again, with Dr. Septinisi, who successfully identified some of the knockouts that did have higher levels of lysine, so up to 7% in the rice grains, simply by knocking out the downstream enzymes in that pathway. So this is another approach to use gene knockouts to modify your end-use quality, in this case in rice grains. Okay, the third example actually is in flowering time, and flowering time is quite a complex trait. So if you see here, this is in rice, there's a short day flowering time pathway and a long day pathway, and there's many genes that are involved in essentially that transition from vegetative to reproductive stage. And so if you look at some of these genes actually are positive regulators and some are negative regulators of flowering, essentially. So if you look at, again, this was mentioned earlier, if you knock out a negative regulator, you can essentially upregulate or induce your desired phenotype. And so the idea here is that we're trying to knock out some of the negative regulators of flowering time so that we can promote early flowering. But it's quite complex because it depends on, again, the day length, so short day lengths versus long day lengths. And there's many other factors that are involved in flowering time. But to achieve this, we actually targeted 6 well-known genes in the flowering time network, and are currently looking at different knockouts of these 6 genes and trying to identify which ones can essentially induce early flowering in rice. Okay, so those are some of the applications. I'll next talk about some of the techniques where we're trying to optimize plant transformation itself. So moving beyond agrobacterium.
Dr. Thompson.
Yes.
Sorry about interruption. I need to make an announcement because we just exceed the scheduled time. We need to reopen the meeting in order to extend its duration. Is it possible for you to stop in here and just 5 minutes later, using same link we provided earlier, can you reconnect again? I am really sorry for this technical issue and interrupting your speak.
No problem. Yeah, we can connect again with the same link.
Yes, thank you. Thank you very much.
Okay. We received funding to essentially test this in several crops, including rice, sorghum, peanut, and cowpea. And we do have some initial results, for instance, in rice. So initially, we tried to do leaf infiltration. It's quite difficult in cereal leaves to infiltrate using standard approaches. So in this case, we used a method where you actually have to puncture the rice leaves and then leave that in a solution of the plasmid DNA carbon nanotube solution for up to 10 days to actually, again, kind of passively uptake the carbon nanotubes into the plant tissues. And what we did is we checked with GUS and GFP reporter genes to identify successful, essentially, delivery of those plasmid vectors into plant tissues. So we did see GUS activity in the leaves. And actually, we also tried this in seeds and mature embryos. So trying to excise the embryo from the rice grain or expose the shoot apical meristem. And again, use carbon nanotubes with plasmid DNA to try to, essentially, it's a fast track approach for plant transformation. And this is, they very quickly produce seedlings, and then we can check if they've been transformed. So we're also testing this in cowpea plants. And this is work from Merve Saglam. So she's a Turkish student in our lab. And she's been testing both carbon nanotubes and carbon dot delivery, into cowpea leaves. So this is using leaf infiltration in cowpeas as well. So it also looks quite promising. We're using PDS and trying to see if we can knock out PDF function in the cowpea leaves. Okay. So that's just, some examples from our lab. And just kind of to conclude, we know CRISPR has tremendous potential to make an impact in crop improvement. We have knowledge about candidate genes and genetic pathways. We can now introduce novel traits, and we can really empower plant breeders to precisely combine desired traits now. And we see this technology rapidly improving. I think, especially in the United States, the regulatory situation is becoming more clear so that we can use this technology for commercialization. But there's still an issue of potentially consumer acceptance. We need to help improve, educate the public on how this technology differs from previous GMO technologies, especially, techniques where it's non-transgenic, it's essentially non-GMO, and that can be very useful moving forward. So with that, I'd just like to thank my team. So this is a current team with myself and Dr. Septuningsi. This is Merve, Sudip, and some of our other students who have performed the work that I showed today. So with that, thank you, and I'll take any questions.
I want to go with that, what's it called? That NASA boss.
Okay. They go by what time?
By 4 this morning. Welcome.
Okay. Thank you for your presentation, dear professor, for your comprehensive presentation on CRISPR-Cas9 technology. We have some questions, so I would like to move on to the Q&A session. My first question will be that, dear doctor, I am Haruna Daniel. How can we get access to these optimized protocols? Can you explain?
Yes, so that's a good question. So, we tried to publish them as quickly as possible, but if anyone would like more information on the unpublished work, feel free to email me. You can go to Texas A&M website and search my name and find my email contact information.
Okay, thank you. The second one, mix of the three questions. Thanks for your presentation. The first one is, in comparison of RMP and agrobacterium-based systems, which one has better efficiency on creating mutations, on which one is easy to apply according to your experience?
Yeah, so I would say agrobacterium transformation is the easiest to use. And so I do recommend people start with agrobacterium. We've had some difficulty with the RNP approach. It's a little tricky to use. So I would say agrobacterium is still the easiest approach.
Thank you. The second one is, combining two mutations of CRISPR in conventional way is not problem, as you mentioned. What about off-target effects that were not identified by owner of registered variety? What's gonna be happen if they have this detected later on in terms of regulation?
Okay, so that's a good question. So I think, in terms of regulation, for instance, here in the US, I mentioned it should be a single deletion, but of course, if there was an off target, that would be a second mutation, and so that's essentially not allowed. So what you have to do is confirm that there are no off target effects in your variety, and so most people initially will PCR amplify any targets that are similar to the one you designed. So for instance, there could be 10 or 15 loci that have similar targets. And the other is just through DNA sequencing, just checking to make sure none of them have off targets. I don't know if I mean, to be very comprehensive, you can do whole genome sequencing at a very deep level to really ensure that there's nothing unexpected in the genome. And I think when companies are commercializing this technology, I think they're being very careful to make sure there's nothing unexpected, because that could be, it can have negative kind of implications on consumer acceptance if later someone finds out that there was an off target that they didn't realize. So I think everyone's being very careful to really ensure that there are essentially nothing unexpected.
Thank you. The last one is coming from researcher Erdem Sefa Şahin. What do you think about that? There was a report show Agrobacterium also inserts its chromosomal segments up to 19 kilobase out of right border and left border into plant genome during the transformation, 1 in every 250 transformed plant. How plant variety registration system deals with this kind of horizontal gene transfer, where single mutation is considered as a non-genetically modified organism?
Yeah, so that's another excellent question, and many things can happen during plant transformation. So, for instance, Agrobacterium, you as was mentioned, may have some segments of DNA integration. Also, biolistics, particle bombardment can introduce some segments of DNA, and so I think everyone, scientists working in this field are being, again, very careful to ensure that their final product does not have any of these extra segments. And so in practice, what we do is we'll develop many lines, gene edited lines. And then if we detect any of these additional segments, then of course we'll discard those samples and continue with others. But I think the important thing is that you have to be able to detect these segments. And so again, we can do PCR with multiple primers across the tDNA or across the entire vector, binary vector. But to be really sure, if you're going to commercialize, I think most people will be doing whole genome sequencing at depth, and so that they can identify, to make sure that the genome is still identical to the wild type genome.
Thank you. The next question is coming from Professor Dr. Sami Doğanlar. Did you test CNT delivery system in cotton? Thanks for your presentation.
Yeah, so that's a good question. So we currently have a project to test CNT in cotton, and we did have a graduate student, testing that using leaf infiltrations. I will say that it's not really been successful yet. I think there's more optimization that's required, especially in cotton. It's been quite difficult, and so I think for every crop it's going to be different whether it can be successful or not. So it is, I mean, theoretically it's possible, but in practice it's very difficult to achieve, and so there's going to be more optimization for each crop.
Thank you. The next question is also coming from, again, researcher Erdem Sefa Şahin. It says, could you explain a little bit more patenting genes? Could you briefly explain how patenting genes can be evolved by developments with CRISPR-Cas systems in gene discovery studies? New allele will be patented or no more patent will be allowed in the same gene?
Okay, so yeah, so another good question. I think, when we're looking at patenting, there's several different types of patents. So some of them, like utility patents, based on kind of the novel function. And so I think in most cases, most people are not looking to patent like natural variation, like it exists in nature. So it's not, in a way, it's not novel, right? Because it's already existing. But if you think about CRISPR technology, it's very powerful. You can actually make, again, you can make modifications that could have potential new functionality. And so you could do, again, I think many people are looking at more into like synthetic biology, again, kind of novel traits, novel gene function. But yeah, in our group, we have not really proceeded with patenting particular genes. I think we're really focused on just developing applications that can go into, especially kind of for public research, to go into, just improving the efficiency of the technology. But yeah, I mean, there are potential applications in that field, I would say.
Thank you. The next question is coming from Ahmet Alperen Canbolat. What is the current state of commercialization and adaptation of CRISPR-edited crops?
Yeah, so at least in the United States, the first CRISPR food that was commercialized was actually using the TALEN technology. So this is the one I mentioned previously. TALENs are using TAL effectors to make the modification. So as far as I know, I think it was the high oleic soybean oil that was one of the first to be commercialized. So that's a gene-edited soybean that produces high oleic acid. And this makes it much healthier as a cooking oil, and also as a, for human consumption, it's high oleic is a much healthier oil. So it's essentially making soybean oil become closer to, say, canola oil in its fatty acid profile. So that's been grown by farmers in the US and actually consumed in restaurants for several years already. But there are other, CRISPR crops now coming online. There's a company called Pairwise. They developed some leafy greens that use CRISPR technology. And these are all in the process, essentially, of being commercialized now. So I think we're just about, at least in the United States, we're about to see a new kind of set of CRISPR crops that are being released and commercialized.
Thank you. The next question is coming from Fikret Işık from North Carolina State University. As someone unfamiliar with CRISPR technology, I'm curious about its effectiveness in editing genes of traits with complex genetic makeup, particularly in relation to traits like yield. Thanks.
Yeah, so that's a common, I think, disadvantage of CRISPR technology in that it's really only effective with, loci of very large effect. So if a trait is controlled by, either one gene or maybe a small number of genes. So I would say right now, there's no clear application for something complex like yield. In the future, once we can get more like multiplex editing, high throughput editing, we could, I think, address more of the complex traits. I will say, like in rice, there are some QTLs that were cloned for yield in particular. And so it is possible to have some single genes of large effect. But in most cases, a simple knockout is not going to improve yield. And so you're going to need some more complicated approach to address something like yield. So that it will take several more years, I think, before we get to that point.
Thank you. I will ask one more question. It's coming from Professor Dr. Sami Doğanlar. Do you have any cotton protocol working cotton cultivars rather than Cocker 312?
Yeah, so cotton has been, is well known to be very difficult for transformation. And so our transformation team has tested a few other genotypes, and they were able to reduce. So most of the genotypes take 2 years to go through the tissue culture regeneration process, and they're able to reduce it to 18 months. But it's still a very, very long time. And I believe they only did that for one additional genotype, but it is quite challenging to work in cotton. And so I will say everyone has the same difficulty.
Thank you so much. And that was our for today. Thank you, Professor Dr. Michael G. Thompson, for your answers and giving your time to our seminar. Thank you so much.
Okay, thank you. And thanks again to the organizers. It's been a wonderful virtual seminar, and I've really enjoyed being part of it. Thank you again.
I'm glad to hear that. Thank you so much. Thank you all for your active participation in today's virtual seminar. I extend my gratitude to our esteemed speakers and the organizations involved for their valuable contributions. Please note that today's seminar has been recorded and will be published on our YouTube channel. I look forward to seeing you all in our next seminar. Thank you once again and have a wonderful day. Thank you so much.
I also would like to thank
Bye-bye.
all attendees and our speakers. Thank you for sharing all these wonderful insights and expertise to Professor Katavelli, Dr. Serkan Tuna, and Professor Thompson, of course. And I also would like to thank USDA-FAS office in Ankara and our team in here, actually. Thank you for everyone making this event. It's wonderful. Hope to see you soon then. I think we can stop. If anyone would like to say something, we can give time. We have a little bit more time if anyone would like to speak. Sami Hocam, would you like to say something?
That was a nice actually event. I'm happy to see Michael Thompson. He's from Cornell too, just like us, me and Anfureri. And then you know him very well. And then please I'll say hello to Septi from me and Anfureri. And I hope to come together sometime, Michael. And maybe we can work together as well.
Sure. Yeah, that'd be great. So good to see you again.
Sure, I will write you email about possible projects.
Yeah, feel free to email.
Okay.
So wonderful. Thank you. Thank you for everyone who just join our event and share their expertise with us, especially to speakers. Okay. I think we can stop in here. We took a lot of time from you, Michael. Thank you very much for your time, especially. So the meeting just extended a little bit more.
Yeah, thanks. I enjoyed it. Okay. Bye-bye then.
Then, yeah. Bye then. Then we can stop.